Hello everyone. I am Halime. I am studying molecular biology and genetics in Turkey. I am member of group Molecular Biology and Genetics Agenastro Turkey platform. I would like to briefly introduce Anameria Berea, which will give us a presentation about computational data science for astrobiology today. Having completed her doctorate in economics and computational social sciences, Anameria is also the first woman to graduate with a doctorate in computational social sciences. She explores the emergent and basic communication pattern in both living and non-living systems, from cell-cell interaction to animal signal, human languages, and metaprogramming languages. She received many awards, some of them the fourth best team in the IRRPA Global Prediction competition in 2019, and DataKind and the European Court of Human Rights Data Ambassador in 2014. She is currently working as a researcher in Blue Marble Space Institute of Science is with us. Hello, Dr. Anameria.
Hi everyone.
How are you?
I'm good. How is everyone today?
Would you like to tell us more about you? Dr. Anameria, you can hear me?
Sure. Yes, there is a little bit of background feedback, but I hope you can hear me. Yes.
Would you like to tell us more about you?
Okay. Yes. Doctor. So my work in astrobiology comes at the intersection of astrobiology and data sciences, or astrobiology and social sciences, particularly in economics.
Yeah. You can, if you want, you can start your presentation.
Okay. What I am going to present today. Okay. So what I am going to present today is work that I research work that I did in combination with the Blue Marble Space Institute of Science in their program, the young. I'm sorry. I can. It's. I can hear a lot of background noise. I can hear myself talking.
Hear clearly.
Okay, so let me see if I can you still hear me?
Yeah.
Okay.
So.
Okay.
You can start.
Okay. So the work I'm going to present today is about the universal constraints to the probability of life that we derived from game theory. And basically here we worked together with the Young Scholars Program at the Blue Marble Space Institute of Science last summer on a series of computer simulations to better understand what are the informational constraints to the probability of life. If we are looking beyond biology, and if we are looking at the information theory, and if we are thinking about living entities in the universe as informational entities as well, can we derive some universal constraints to the probability of life on exoplanets if we are using some of these computational tools or data science tools, such as computer simulations or game theory? To begin with, since probably not everyone is familiar with game theory, I would like to talk a little bit about what game theory is. So game theory studies strategic interactions between different interacting entities. Originally, these were defined, or they were studied in the economic space. So these were economic interactions, but they are not just economic interactions. Game theory is more or less a branch of mathematics. But another way in which game theory has been used is to study cooperation versus competition behavior. And cooperation and competition behavior, we can see that not just in humans, but we can see that pretty much in any living systems, in any living forms. So are we to expect that if we are looking for life on other planets to see also this kind of cooperation versus competition type of behavior? And some examples here are let's say the zero-sum games. So probably this is something that you are more familiar with, or you have heard more about, where in zero-sum games, basically the players, if someone wins, the other one loses. Another game theoretic setup that you're probably familiar with, or maybe you have heard of, is prisoner's dilemma, and I'll talk in a minute about what prisoner's dilemma is, and so on and so on. So there are many games that have been derived mathematically, and these are not necessarily games as we think of them as games of chess or such games of tennis, or these are strategic games that involve basically at least two parties that are interacting with each other in order to achieve some individual outcomes. The first general mathematical formulation for game theory came from John von Neumann and Oskar Morgenstern in a paper that they published in 1944. So this is not a new theory, or it's not a new branch of mathematics. It has been for a while, and it has been particularly used in, as I was mentioning, in economics, in other social science type of setups, but it has also been used in biology, and I will get into more details shortly. Actions that the actions in a game theoretic setup are basically informed by expectations and interactions between those entities that are playing the games. And as we will see in a moment, you don't have to be a human in order to in order to be in a game theoretic setup. Why are we using game theory? Because of its robust mathematical formulation. It can be it can be unbiased when we are talking about different living systems or different biological entities that are in that particular setup, which means that basically if we are to think about living systems or living life forms on other planets, we are not bound by what we understand biology and life to be on Earth, right? So we are moving towards a more general mathematical formulation about how living systems would interact regardless of their biology. To give you an example of such a game theoretic type of setup or thinking. Historically, when Cortez arrived in the New World, and he was faced with the population that was native into the New World, so this was on the shores of current Mexico. The game theoretic decision that he did was not about to leave no option to the soldiers, but about the message that he was sending to the Aztecs. So the setup in this, the historical setup here is that once these ships arrived on the shores of current Mexico, they were faced with either moving on to actually conquer the Aztecs or going back. So what Cortés chose was to burn the ships in Veracruz in Mexico. Why did he do that? So what's the rationale behind it? So probably it's, if you are thinking about this historical example, it's not, it's not a great example that we would sympathize with, right? But let's think about this example only from the game theoretic point of view and not from the social point of view, okay? So if we are thinking in this example only from the game theoretic point of view, we can say that any commander who could be so confident as to willfully destroy his own option to be prudent if the battle went badly for him must have had some good reasons for such extreme optimism. So the signal that he sent to the Aztecs was that they were so powerful and confident that they were willing to have no option of return. So it cannot be wise to attack an opponent who has a good reason, whatever exactly it might be, for being sure that he cannot lose, right? So again, if we are thinking about this example out of the historical context and of the social and cultural implications, which of course are not the most, the most fun or happy ones. But if we are thinking about this only from the game theoretic point of view, it's basically a signal that one of the players is sending to the other players, that they are so confident in their ability to win that they leave no room for turning back. And what happened is that indeed the Aztecs, when they saw the burning ships, they were terrified of the new incoming army, and they lost in the battle. So this is related to the idea of the economics of collective action. And the economics of collective action is not, again, just about humans or just about the history of us on this planet, but it's something that we can apply and think about in all living systems. If we are to dig down into this example that we had with Cortés burning his ships at Veracruz, what are the rationales, right, behind this? What are the rationales behind these types of actions? So the soldiers are not motivated only by self-interest, right? So they could run away in battle. If we are thinking about the Spanish soldiers, right? So why did Cortés burn the ships? The soldiers were also motivated by what other soldiers would do and what they expect others to do. So if some soldiers would flee and turn around with the ships, then others will follow. Even a quite brave soldier may prefer to run rather than heroically but pointlessly to die trying to stem the oncoming tide all by himself. But without the option to go back, then all remaining option was just to move forward. So the retreat is not only made physically impossible, but also economically impossible. So it means that the cost of retreat is at least equal or higher than the cost of fighting, which meant perhaps death, right? So if we are thinking about the costs of action in terms of costs of fight or flight, which we see in all living systems, and we are thinking also about the benefits of fight or flight in all living systems, then we are moving towards thinking about this idea of the economics of collective action in other biological species as well, right? Do you fight or do you do you leave? Is it fight or flight? It's something that perhaps we can find in other living systems or in other life forms on other planets as well. Some other example, as I was mentioning, zero-sum versus non-zero-sum games. These are some other examples from game theory. In nature or social systems, we rarely find zero-sum games. So the game where we are thinking that someone always wins at the expense of someone else, that someone always loses, someone else and the other wins. It's actually more of a mathematical formulation, and it's not something that we find in nature. In nature, we are more likely to find non-zero-sum games where both players win or lose. A zero-sum is usually a fallacy because one does not gain what the other loses or vice versa, while the non-zero-sum games allow for individual competition and cooperation in a system to lead to emergent patterns at the system level. And it's something that we are also to expect into other life forms that we can see on other planets, because again, this is about the behavior of the individual rather than tied to the biology or the environment that we might find on other planets, and that we currently don't know how that looks like. Another example that I was giving of a game was the prisoner's dilemma. So in prisoner's dilemma, rational behavior leads to socially unbeneficial outcomes. So how does this work? So if you have two thieves, right? Two partners that were caught, let's say, stealing something, and then they are being interrogated by the police, and they have the option to cooperate with the police or to defect. So again, here is about cooperation versus defection. And you can have different payoffs or different outcomes if you either cooperate, either if you defect. And what has been shown in this game was basically that in general, the parties, the two entities that are playing the game, usually choose to defect, which means that rationally it would mean that you are more inclined towards individual outcomes, to individual behavior than to the group behavior. And if you are a little bit more familiar with game theory, this is what is called sub-Nash equilibrium. But in an evolutionary setting, where you play these games repeatedly, which we can see in nature because it's not just a one-time, one-instance type of situation, and we add the evolutionary aspect that we see in biology, then the outcomes are actually quite different. So if we are thinking about evolutionary game theory, this originated as an application of mathematical theory of games to biological contexts, arising from the realization that the frequency dependent on the fitness of the organism introduces a strategic aspect to evolution. Evolution itself is treated by evolutionary game theory does not necessarily mean biological evolution. It often can be understood as a cultural evolution as well, where this refers to changes in beliefs and norms over time, if we are thinking about humans. But also think about this idea that we see cultures not just in humans, but we see cultures in big apes, we see cultures in cetaceans such as dolphins and whales, and we can certainly apply evolutionary game theory in some of these more advanced species. The rationality assumptions underlying evolutionary game theory are, in many cases, more appropriate for modeling of social systems than those assumptions underlying the traditional theory of games. So if you think back here about just one instance of prisoner's dilemma, then the outcome here is always about individual benefit rather than the group benefit. While if we are thinking in terms of evolutionary game theory, then the outcomes are different, and we'll see in a minute how those are. Evolutionary game theory was, with applications to biology, was first developed by Fisher in the genetic theory of natural selection all the way back in 1930. So again, just like the underlying mathematical theory under game theory, these are not very new ideas. but the way we are applying them now are quite new, and we'll see in a moment. So the original genetic theory of natural selection was the attempt of Fisher to explain the approximate equality of the sex ratio in mammals. So how does this work? If we are thinking about extinction risks that are based on the fitness of the individuals or of the species, the question here is, why is that the sex ratio is approximately equal in many species where the majority of males never mate, right? So this is a large evolutionary question in biology. In these species, the non-mating males would seem to be excess baggage carried around by the rest of the population, having no real use. But Fisher realized that if we measure individual fitness in terms of the expected number of grandchildren, then individual fitness depends on the distribution of males and females in the population. When there is a greater number of females in the population, then males have a higher individual fitness. When there are more males in the population, then females have a higher individual fitness. The fact that individual fitness depends upon the relative frequency of males and females in the population introduces this strategic element into evolutions, and we can think about large evolutionary scales in terms of game theory and evolutionary game theory. So this brings us back to the idea, what if we take the classic prisoner's dilemma and we introduce this evolutionary aspect to it? What does it mean to introduce the evolutionary aspect to prisoner's dilemma? It means that we will have multiple players with multiple rounds instead of just two players that play this game only once. And we end up, after repeated games, is that we observe the evolution of cooperation. So basically, if we have an opponent and yourself, that you are playing the prisoner's dilemma, and we have a payoff matrix where you cooperate and you have an outcome of 1 or a payoff of 1. And if you defect, you have an alpha parameter that can be between 0 and 1. If one of you defects and the other one cooperates, right? So the strategies are different. But if the strategy is the same as that you both defect, then the outcome is 0. So it's non-beneficial for both parties. So the only outcome that remains is that both parties cooperate. And we'll see in a minute how this works in this evolutionary setting. So basically, if we are introducing these games into a simulation where we have a population of cooperators and of defectors, and cooperators are with red and defectors are with blue. And we are tweaking for different defection awards for those alpha parameters, and we are also tweaking for the population ratio, that how many cooperators versus defectors we have. Then we have this kind of setting of evolutionary games, right, where we have these agents in the simulation that keep on playing in this evolutionary setting, and we are observing the outcome. So what we can observe after repeated games in a population of living systems or entities, that some of them are cooperating, some of them are defecting, is that, of course, if we have a higher parameter of the initial cooperation ratio, so we have more cooperators in the beginning than defectors, then we do arrive at a population that is fully cooperating, this color blue. If we are also tweaking for the defection award, so this means how much payoff do I get if I defect versus if I cooperate, right? If I compete versus if I cooperate, then we also observe that the higher the defection award, then of course the population is going to be more competitive than cooperative. And we'll see some more examples in a minute. So some of the observations from this kind of generic evolutionary prisoner's dilemma is that There are a few assumptions about the initial conditions, right? So those initial conditions are, again, how much of the population originally, of our population of biological entities, is cooperating versus defecting, and what is the parameter for the defection award. Then there are very specific ranges of cooperation and defection awards that allow for the system to evolve. What we'll see in a minute is under which combinations of these two parameters we will end up with a system where we have a non-equilibrium system that keeps on evolving versus where we have systems that reach an equilibrium and die. Most parametric combinations from this kind of generic prisoner's dilemma actually lead to stationarity, meaning extinction. So in general, we observe that whether the agents here are cooperating, I'll run this again. So whether the agents here are cooperating or competing, usually it leads to stationarity. So this is not stationarity as we see here, because the system keeps on evolving and we have variety in the system. But this one leads, for instance, it's a system that dies, that leads to fully competition. And the other way, we can see that the system will end up settling into a fully cooperative system, which is also a system that dies because it leads to this kind of stationarity. And stationarity means extinction. Now, okay, we have this theoretical framework that is both mathematical and computational because we put all this into these simulations. And how do we apply this, though, into an actual biological setting? So if we are thinking about how we can apply evolutionary prisoner's dilemma to a game of viruses, and we can take the example of viruses again so that we are not specifically bound to any biological species that we know on Earth, but we have something that is more generic, and we know that, for example, viruses are not necessarily even thought of being necessarily living systems. But if we are applying evolutionary prisoner's dilemma into the setting, into the context of viruses, again, we are removing a lot of the bias that we have when we are thinking about different species and how they behave on Earth, right? So we are moving into a more general, universal framework. This particular research is based on this paper, Evolutionary Stable Equilibrium and the Extraterrestrial Interactions, by Scott Colby and Megan Van Althuis at State University of New York in Morrisville. And in this research, natural selection can function as a player by selecting behaviors and genotypes from viruses, right? So in this setting, it's natural selection that is one of these players in prisoner's dilemma. The stability of an equilibrium strategy can be tested by introducing a small mutant or invading or defecting population. So we will see in a minute the kind of gene that is being introduced that functions as a mutant or as an invading or defecting population. If mutants are less fit than those playing the equilibrium strategy, then the equilibrium is an evolutionary stable strategy. And human-like cognition, right, is not necessary for game theory. So that's why we are looking at virus games. We don't need human type of decision-making. We don't need a species type of behavior. It is general and universal enough that we can potentially apply this to extraterrestrial living systems that currently we don't know their biology or how they look like. And how do we apply this? We took the observations or the research from this paper and the outcomes from this paper, and we actually introduced them into an agent-based model of natural selection and cooperation. An agent-based model is what I was just showing you before. It's a simulation where we have different types of agents for which we are looking at how individual interactions, how they emerge into larger patterns into the whole systems, right? So it's a methodology specific for studying and understanding complex systems and studying and understanding emergence at the level of the system from individual interactions of the agents. Again, in this kind of model or simulation, the virus strategies are chosen through natural selection. And when multiple viruses compete for the same resource, so basically they are competing for invading a cell, they may exhibit one of the two phenotypes. What are these two? One phenotype is to cooperate by manufacturing an abundant product, and the other phenotype is to defect by making some product and using a lot of the products that another virus has made, right? So again, we have viruses that are competing for cells and that they are manufacturing more RNA for that cell. The fitness levels in this game are estimated experimentally using the data from this paper by Turner and Chow, and we normalize those fitness levels to 1, right? So we actually took actual data from laboratory experiments and we introduced that into the simulation so that we can give more specificity to the general mathematical framework that I was showing you before. This is the payoff matrix in virus-virus prisoner's dilemma from the paper that I was just mentioning by Turner and Lin. And we have virus 1 that is playing against virus 2. They are competing for one cell to invade one cell. And we have two different phenotypes, Phi6 and Phi-H2, that are about cooperation versus defection for that cell. And the numbers that you see here are the ones that have been experimentally observed into the laboratory. We took these numbers and we introduced them into a repeated games in an evolutionary setting. Some comments on this virus-to-virus prisoner's dilemma. These empirically derived fitness payoffs imply that viruses face a prisoner's dilemma game with a Nash equilibrium for the phenotype H2H2, right? So if we go back here, this is, again, as in classic prisoner's dilemma that I was showing you earlier, the viruses, if they would play this game just once, they would settle for both defection. So this would be for individual payoff and not for the larger collective payoff. Okay, so it would be more for for defection and not for cooperation. So again, just like in the general prisoners dilemma, they would settle for some Nash equilibrium. The defector strategy that is employed by the gene by the phenotype H2 provides an advantage over the Phi6 strategy during the co-infection, regardless of the other virus's strategy. So in other words, if we are looking at the defection phenotype strategy of the H2, that one strictly dominates the cooperation, the Phi6 strategy. This allows for Phi-H2 to invade the population of Phi6, but as Phi-H2 becomes more concentrated within the population, its overall fitness decreases, as we were showing in the theory of Fisher that was published in 1930. Nevertheless, even as high Phi-H2 concentrations, it is better to be a Phi-H2 defector. So this explains why Phi-6 phages disappear from the mixed populations of Phi-6 and Phi-H2 phages. So what does this mean? It means that these cooperative genes are slowly disappearing from the overall population, leaving the defective and competing phages. Sorry, I shouldn't say genes, phages. Yet the resulting Phi-H2 population is 17% less fit than the members of a purely Phi-6 population. So this seems at odds with the principle of survival of the fittest. However, recall that the natural selection selects individuals, not populations. So the relevant question is, what phage will provide the greatest fitness in a given environment? In order to find that out, we put all this information and data into an evolutionary game theory. So in an evolutionary setting, we set the defection award to be 0.83, which we observed to be the sub-Nash equilibrium in laboratory experiments. We set initial cooperation to 0.5, which basically represents random chance, right? 50-50. The population becomes extinct almost immediately if we are running this kind of scenario. But now let's look at several other scenarios, right? That's why we have all these simulations and computational tools. So for a better alternative, we explore all the parameter ranges for initial cooperation. We do what we call in simulations a parametric sweep. We allow for 3 types of defection awards. 0.83, the sub-Nash equilibrium one observed in experiments, for 1.99, and the relative defect award when the other defects and you cooperate of 1.34. We observe that when the system does not lead to equilibrium, or when it leads to equilibrium over a longer time, right? So we run all these possible scenarios, all the possible combinations of parameters that we can, and from the outcomes, we take only those outcomes when the system does not settle, right? When the overall system does not lead to equilibrium, whether that equilibrium is about cooperation, whether that equilibrium is about defection, it doesn't matter, it would die, right? As long as it doesn't lead to equilibrium, we take those simulations and we observe which were the original combinations of parameters that led to the evolution of the system. So some observations from the simulation experiments. What we observed is that sub-Nash equilibrium always leads to extinction. It's kind of expected, right? Because the sub-Nash equilibrium is always about defection, is always about competing. The high defect payoff, when the 2 populations play different strategies, leads to islands of survival from one population within the mass of the other population. So increasing the initial cooperation ratio leads to more islands, but not longer survival. So if we are looking at this scenario over here, we can see basically these islands of survival for some of these agents in the system. So we can see under certain situations more islands, but shorter time of survival. Now, how can we look at more, how can we increase the survival time and see how we can get into more, systems that evolve more and last for longer time? So we looked again at different other scenarios. And if we are looking at the different parametric initial conditions, such as low initial cooperation versus high initial cooperation, what do we observe? So if we are looking at the relative defect payoff, that one of 1.34 that we calculated, that we observe that it leads to non-equilibriums in the system. So let's look first at low initial cooperation. And we can see in this simulation how basically, even if we have originally a population that is more inclined to defect and not cooperate, but we have just the right defection payoff, then this leads to an evolutionary longer surviving systems. Now, let's look at high initial cooperations of the opposite of that. So if this one basically starts with more defectors, if we are looking at this other one, sorry. Okay. Just give me 1 second. If we are looking at the other one, okay, just give me 1 second to start it. Okay. Okay, so this one is for the initial low initial cooperation, and I will let this run until the end so that I can start the other one. Okay. Okay, so in the other one, we basically start with high initial cooperation, so we have a population of blue agents that it's predominantly blue. But again, looking at the relative defect payoff, which is 1.34 that we calculated, again, we see non-equilibrium system. So what does it mean? It means that basically if we have the right parameter for the payoffs for defection, it doesn't matter if the population originally is inclined to be more towards competition, or it's inclined to be more towards defection, or so if it's inclined to be more towards competition or cooperation, right? If it's inclined to be more towards defection versus cooperation. So this is quite an important result, because it shows us that regardless of the makeup of the population, with the right parameter for the defection payoff, we can achieve evolutionary longer survival systems. So some of the conclusions from this research, what are the conclusions that we can draw so far? We can see that cross-disciplinary methods, such as evolutionary game theory, agent-based models, are quite powerful for us to understand the limits of systems and interactions, such as we see in living systems. And they can easily be extended into astrobiology, where we need to think about how to remove the bias that we have about the living systems on Earth, right? Because we don't know how life on other planets, how it might look like, whether they are viruses, microbes, or more advanced life forms. We don't know the context or the situations in which that those life forms evolve and thrive on those exoplanets. But we can find, or we can think about larger theoretical frameworks, larger computational and mathematical frameworks from which we can derive scenarios where we have some of these biases about biology on Earth. We can have some of those removed. And they can explore scenarios that we currently do not even exist in our reality or the way we understand biology on Earth today, right? So the whole idea here is, if we are thinking more about what we expect in terms of evolution, in terms of interactions between and behaviors between different entities or living systems on other planets, then we are more likely to think about what it's possible to find and what are the limits. From the informational theoretical point of view, what are the limits of what we can find on other planets? And some useful links, if you are interested in exploring more some of these interdisciplinary approaches between mathematics, computational sciences, economics, and astrobiology, I actually have a website, complexeconastrobio.org or .com, and I also have a blog on data science and astrobiology, which is hosted by my university at George Mason University, that you can subscribe to, and where I post more ideas and more research that is along these lines. This is all I had for you today. I don't know if this is if I can take questions, if I can answer some questions or some comments. But I hope you enjoyed this presentation, and it makes you think about astrobiology and life on other planets in a little bit of a different way. Okay, thank you.
First of all, thank you for accepting our conference invitation and for your wonderful presentation. We have a questions.
Okay.
Someone audience asked that, do you think these programs illustrate the evolutionary process correctly, or are there differences?
Yeah, so this is a very general framework for understanding evolution. Obviously, all evolutionary processes are not the same. And we have different evolutionary processes for different environments and species. But we are looking at, first having a more general framework and general umbrella for how we understand evolution. And then we can apply this into specific situations.
Thanks for answer. Another questions from do you think a planet consisting entirely of computers could have been formed in the universe?
Yes, it is possible. Yes, so a planet formed of computers. In order to get a planet formed of computers, you would first have to have biology that leads to those computers, because usually technological innovation or technological evolution follows the biological evolution. We don't know of any processes so far where we have entirely artificial systems without having a biological start for those artificial systems.
Thanks for answer. Someone audience asked that do the scores in the payoff matrix change if the interaction between the organisms change with time?
It is possible indeed. So in some of the evolutionary game theory, we can also tweak for that alpha or for that defection award, that it changes with every game, where the players, when they are interacting with each other and based on their past interaction, actually change their defection award. So yes, that is possible.
I have a two question. First of all, can mutation in viruses be predicted in this area?
What do you mean by notation?
Yeah. I have one more question.
No. Okay, just can you repeat the question, please?
Yeah. Can mutations in viruses be predicted in this area?
Mutations. Sorry, I misunderstood.
Yeah.
It's hard to. Yeah, mutations are very difficult to predict. They are difficult to predict in current biology, in our current systems. It would be even more difficult to predict them for some organisms that we don't know how they look like. It doesn't mean that in the future, at some point, we won't be able to, but currently we don't have that kind of theoretical understanding or framework for predicting mutations.
Mm-hmm. This is pandemic period is important.
Yes.
I have one more question. What would it be like if eukaryotic chromosomes had two or more centromere regions? Or why do we have one centromere region of two? So how would the history of life change? Or can we calculate it?
Um, Again, it's a little bit difficult to predict how life would look like if we had more chromosomes or different types of chromosomes. We don't have right now enough information about how these interactions would play out on the long run.
Because many diseases not accurate because of unfortunately we have reached the end of the period. Thank you for your wonderful presentation.
Sure, it was great to be here. Thank you for the invitation.
See you. Take care.
Okay.
Our next part is with Dr. Charles. We have a short break and see you later.
Hello, Cardalin. Cardalin, you're on mute, by the way. You're on mute.
Thank you for the heads up. Okay, I'm starting again then.
Got it.
I'm a member of Agenastro team and I'm host of this session. Our guest for the session is Professor Charles Cockell. He's a professor of astrobiology at the University of Edinburgh. He's going to talk about his work on using bacteria to do mining in space. How are you?
Yeah, very good. Thank you very much, Cardalin, for inviting me. I was just watching the last two talks and it's been a really outstanding conference. So thank you very much. So I should say, Mihaba Burada Olmak Güzel. That's my attempt to say it's good to be here. And now I'm going to try and share my screen.
Yes.
Share.
Can you please give us a brief talk about yourself before you start your presentation?
Yeah, sure, I can. That looks like it's working. Good. Yeah. So before I begin the talk, I'm Charles Cockell. I'm professor of astrobiology at the University of Edinburgh, and I study life in extreme environments. And today I'm going to tell you about some space experiments with the European Space Agency. And I also study the habitability of other planets. And I have a great deal of an interest in the human exploration of space as well, and the future of human societies in space. I did my PhD actually in medical molecular biology, nothing to do with astrobiology at all. When I did my PhD, astrobiology did not exist. It was not possible to do astrobiology. So this actually came later in my career. I worked at NASA for 5 years on life in extreme environments. That's actually when I became an astrobiologist during my postdoc period. And then I came back to the UK. I worked for the British Antarctic Survey, doing microbiology in Antarctica for 5 years. I wasn't in Antarctica for 5 years, but I was working at that institution and went down to Antarctica a few times. And then I moved to the Open University, where I worked on astrobiology and life in extremes. And then I've been at Edinburgh, gosh, now for 10 years, where I set up the UK Centre for Astrobiology when I came here. And we've been doing all sorts of different astrobiology projects here at the University of Edinburgh. And today I want to tell you about one particular set of experiments. So if you're interested to talk about careers in astrobiology, I'm very, very happy to have these discussions either today or actually at any other time. But if you have questions about my journey, if you like, in astrobiology or any career questions, please do ask at the end of the talk. I'm very happy. We don't have to just talk about the science behind my talk. We can also talk about astrobiology generally, given this is an astrobiology conference. So I'm going to share my screen, and Cardula, can you tell me if that is working? Hopefully that's working. Good. Okay. Thank you very much again.
Yes, we can hear you. Okay. You can start.
Okay, good. Thank you very much. Good. So I want to tell you today about some work that we've been doing on the International Space Station recently, as I say, with the European Space Agency, developing microorganisms for use in space. Now, microorganisms are extremely versatile organisms, extremely versatile creatures.
Professor, I'm sorry to interrupt, but the slide doesn't move.
Oh, really? Is that not changed?
No, it doesn't change.
Huh.
Can you try to share it again?
Yeah, let me try again.
Okay. Yes, it's working now. Thank you.
Okay, let me make it full screen. If it doesn't work, I'll just go back to sharing, without the full screen. So has that moved forward one?
It doesn't move. No, it doesn't move now. But it did move.
It doesn't move.
No, now it doesn't move.
Huh, that's strange. It must be something to do with full screen.
Now it moves.
Now it's moving. Should we, let's
use
just,
it like that?
okay. That's very, very strange. I'm not sure why that is.
Me neither.
Let me try one more thing. Sorry, everyone. Just going to try one more.
Sure.
Let me try sharing the whole screen. Does that move?
Yes, yes.
Okay, let's stick with that then. So I was sharing the application rather than the screen. So this seems to be good. Okay, good. And let me know if anything goes wrong while I talk.
Of course, of course.
Okay. Okay, so microorganisms are extremely versatile. And of course, in some sense, they are nature's natural engineers and factories. They have been processing materials. They have been mining rocks for over 3.5 billion years. And today, rather than a sort of fundamental science talk, I want to talk more about the applied use of microbiology to help support the long-term exploration and settlement of space. And here are some examples of the way in which microbes can be used. For example, in life support, to produce oxygen. We might use microorganisms to produce drugs and other pharmaceuticals to support humans beyond Earth. We might use microorganisms to create biofuels, to supply all sorts of industrial processes and fuel requiring steps in our presence on other planets. We might use microorganisms to break down rocks, to make soil. I'm going to come back to this in a moment. And we can also use microbes to produce plastics and other products. And this might be augmented, for example, by synthetic biology or genetic modification. And what I'm going to talk about today is some experiments we did in space to look at the use of microorganisms in biomining, biological mining. You may not be familiar with this, but about 20% or so of the world's copper and gold is today extracted from rocks using microorganisms. The microorganisms are used to break down the rocks, to oxidize them, and release these economically interesting elements in this process called biological mining or biomining. And the reason why we use microbes is because it allows us to get away from using toxic compounds, like, for example, cyanide compounds, to extract elements from rocks. It's more environmentally friendly. And of course, like I said a moment ago, microbes have been mining rocks for 3.5 billion years. So they know how to do this. And instead of trying to recreate mining processes for ourselves, maybe we can just get microorganisms to do mining for us in space, just as we do on the Earth. And I've written down here, these capacities can be augmented by synthetic biology and genetic modification. Now, I'm not going to talk about the human microbiome, which is illustrated in this slide, but I simply wanted to point out that human beings are an inescapable part of the human exploration of space. The human body is about 50% mammal cells and 50% microbe cells. So, weird thought for the day I've written up here. Humans are actually exoskeleton mobility suits for microbes. Half of the cells in your body are microbial. So you're not actually a human being at all. You're a microbe-human hybrid organism. And we are increasingly realizing the inseparable nature of this human microbiome with human physiology. So the point I want to make is that when we go out into space and establish settlements on the moon or Mars, microbes are coming with us, whether we like it or not. And therefore, we might as well employ them to do useful things rather than seeing them as passive hitchhikers that just happen to come along to the moon or Mars with us. We should think about how we can actively employ them to do industrially useful processes. Today, I want to focus on one particular use of microbes, and that is to transform rocks or regolith on other planetary bodies into useful materials. And this is a paper now from actually 10 years ago, 11 years ago, but it has this figure in that I still find useful. And it's a demonstration of the different ways in which microbes might be used to process rocks or materials on other planets. For example, we might use microbes to break down rocks and soils to extract useful elements in biomining. That's going to be the focus of the rest of my talk. But I want to also draw your attention to the fact that we can use microbes to break down rocks and add carbon and nitrogen into rocks to make soils, maybe for growing crops in extraterrestrial environments. We might also use rocks and regolith to release nutrients in life support systems. Rather than taking all the nutrients you need to the moon or Mars to support algae and cyanobacteria, why not use local regolith and rock material to support microbial growth? We might also use microbes to remove atmospheric dust through microbial crusts. And we can also use microbes in fuel cells, microbial fuel cells using local regolith. And of course, on Mars, as you heard earlier today, the interaction of microbes and rocks leaves behind biosignatures with which we could use to look for evidence of past life on that planet. So these are the diversity of ways in which microbes might be used to carry out economically useful or practically useful processes on other planetary bodies. And today I want to tell you about two experiments that we flew. I'm going to focus particularly on BioRock that we flew to the International Space Station. And I hope that as well as the science, this might also give some of you ideas about how to plan experiments to fly beyond Earth, how maybe you could organize Turkish space experiments and think about how you could do similar types of experiments through these growing number of space opportunities. Both of these experiments were to study how we can use microbes to process rocks and do economically useful things beyond the Earth. Now, you might say, why would we care about microbes growing in space? What's different about the space environment? Well, one of the big differences is differences in gravity. And gravity is known to affect microbial growth. Now, most microbes are very, very small. They're about 1 micron in size. And we think that most microbes cannot directly detect gravity. They're too small to directly detect gravity across the length of a microbe simply because of their small size. But what gravity can do is to affect processes like convection and mixing and sedimentation. In other words, it changes fluid dynamics. And that change in fluid dynamics indirectly affects the growth environment for microbes. So this was a study done by Kim et al., published in 2013, where they studied biofilm growth in space. And I just wanted to use this to illustrate to you why we might hypothesize that gravity would affect the way in which microbes grow in space. And they found that when you grew microbes in space, you got these unusual column and canopy shaped biofilms you can see on the right hand side here in the red ellipse, compared to these mushroom shaped biofilms on the left hand side. And the hypothesis here is that lower microgravity or microgravity is preventing fluid mixing and thereby changing the growth conditions for the microbes and causing differences in microbial growth. So one of the things that we might wonder is, does gravity influence the way in which microbes interact with rocks? And we could hypothesize that gravity will change both the way in which organisms interact with rocks and what they then do with those rocks, particularly with respect to biological mining or biomining. And this was the objective of the BioRock experiment that we sent to our space station in late 2019. So this is a collaborative project, just to talk a little bit about the complexities of organizing space experiments. They're quite difficult to organize. This experiment took 9 years to organize. Nowadays, it's easier to do these experiments more quickly, but this was a collaboration with the European Space Agency, Kaiser Italia, who built the hardware that I'll show you in a moment, and our academic collaborators in Belgium, Germany, and Aarhus University. And what we needed to do is, the first thing we had to do was design a miniature bioreactor to do our experiment to study the way in which microbes interact with rocks. And this was our first challenge. And so we spent 7 or 8 years building this tiny bioreactor, which is about 9 kilometers. This is about 9 centimeters long. The reason why I'm distracted is because I've got one on my desk, but I think it's difficult to show you. I don't know whether you can see that or whether you're seeing the image of me in the camera, but this is a bioreactor that I'm showing in the camera. And this bioreactor is a small container. It's got 2 culture chambers. You can see the 2 culture chambers here. And inside this culture chamber, we've got our piece of basalt rock. This is the material the microbes are going to mine. And then in the back of the container, we've got holes containing the medium and the fixative. And you can see these reservoirs down here. And so we launch these into space with the microbes dried down on the basalt rock. And then when they get to the International Space Station, I'll show you in a moment, we inject the medium, and the medium allows the microbes to grow. The experiment runs for about 20 days. And then at the end of the experiment, we inject fixative, and the fixative kills the microbes and stops the experiment. And that then allows us to bring the experiment back down to the Earth and study the leaching of the elements, or the biomining of the elements. So this excuse me, took some time to design and develop. And you can see it's quite simple, but in fact, it's not as simple as you might think, because of course there are safety concerns with launching apparatus up to the International Space Station where you have astronauts. So you have to have a double containment system, and you have to have various other pieces of safety apparatus within the container that makes it safe for use on the International Space Station. So this took some time to develop. We studied 3 microorganisms in the space environment: Sphingomonas desicabilis, Bacillus subtilis, Cupriavidis metallidurans. And we selected these organisms based on a number of considerations. We wanted microbes that naturally came from rock environments and that naturally interact with rocks. We wanted them to be desiccation resistant, because remember I just told you that we launched them up to the space station in an inactive state, because we only want them to be active during the course of the experiment on the International Space Station. So they needed to be desiccation resistant. We wanted microbes that could be easily studied in the laboratory. In other words, we could grow them easily and understand their effects. And we also wanted to study them all in the same medium. So we used R2A as the medium. And the reason why we did this is because there are very few experiments that study the growth of multiple microorganisms in the same apparatus and in the same conditions. And if you look at the literature on space microbiology, one of the things you'll notice is that there's an enormous number of types of apparatus and types of growth medium. It's very difficult to compare results from one experiment to another. But in this experiment, we wanted to study growth of these 3 organisms in identical medium so we could make comparisons. And I'll show you some of that data in a moment. So these are our 3 microorganisms. And this is just to show you the small bioreactor with the pieces of rock inside the bioreactor. On the left-hand side, you can see a square of this basalt rock that goes into the bioreactor. Now, what is basalt? Basalt is basically what Mars is made from and most of the moon is made from. So it's a very common material in the solar system. And if you wanted to mine rocks elsewhere, this is the type of material that you would find at least on the moon or Mars. Asteroids are slightly different. And of course, there may well be rocks with higher concentrations of elements. I'll come back to that in a moment. But this is a very common material in the universe, and we might want to think about mining materials from these different types of rocks. So that's our basalt. Now, as I say, this is a relatively simple experiment, but it's amazing how complex it gets. So we've got 3 different organisms, which you can see here. We've also got a negative control in the gray, which is basically the rocks without the microorganisms. We've got 3 different gravity conditions: microgravity, essentially no gravity; simulated Martian gravity, because we wanted to see whether we could simulate mining on the surface of Mars; and also simulated terrestrial gravity. Now, you might think, why would you fly into space and simulate the gravity on the Earth? Well, the reason for this is that on the International Space Station, it's not just the gravity that's different. There's also slightly different radiation environment. There are small vibrations in the International Space Station. So it's really important to have a control on the International Space Station that simulates one gravity that you can then compare with terrestrial gravity on the ground. And this basically allows you to check whether the ground-based experiment gives you the same result as the 1G control on the International Space Station. So it's an internal control, if you like. And then, of course, we want to do all of these experiments in triplicate because we want good statistics. So you can see that very quickly, we end up with 48 of these growth chambers. So a quite simple experiment with 3 microbes turns out to be quite complicated. And if any of you out there are thinking about doing a space experiment, my one piece of advice I've learned the hard way is keep the experiment as simple as possible and try and keep the apparatus as simple as you can. So we flew our experiment onto the International Space Station through SpaceX Commercial Resupply Mission 18. This went up, as I say, in 2019. We went to the Kennedy Space Center to prepare our experiments, to the International Space Station Processing Facility, which is where you prepare your experiments and then hand them over for flight to the space station. And this is our experiment being launched on a Falcon 9. This is very exciting, by the way. I mean, I try and not get too, over emotional about this because this is about doing science. But when you spent 9 years planning an experiment, it is really exciting to see it finally taking off and to actually hear and see the roar of a rocket launching into space with your microbes on board. So this is the Dragon capsule docking with the International Space Station with our biological mining experiment on board. And I should also say this was the first mining experiment to be done in space, which also made it additionally exciting. So for this experiment, we used the cubic centrifuge, which is essentially an incubator that contains in the middle a centrifuge. And you can see Luca Parmitano, who was an Italian astronaut assigned to our experiment on ISS, putting the reactors that I showed you earlier into the cubic centrifuge on board ISS. And we used 2 of these centrifuges on the space station. So this is just to show you a cross-section through the incubator. It's not very interesting, but I just wanted to show you that one of these cubic centrifuges has 6 containers simulating microgravity. One of them has 6 containers spinning and simulating Earth gravity. The other one has 6 simulating Mars gravity. And these blue containers are the microgravity containers that are not subjected to spinning. So they're essentially sitting in the microgravity or practically zero gravity conditions in space. And we've got Martian and Earth gravity being simulated in the interior of the cubic centrifuge incubator. You'll also see why it took so long to design these incubators, these tiny growth chambers, because of course they have to be connected into the incubator and from which we can get telemetry or information about the status of these growth chambers. And then finally, after 21 days, after fixating, our experiments were put back in the Dragon capsule and returned to the Earth, hopefully without seawater. And we go and collect them in LA and then do the experiment. Now, while we were flying this experiment in space, at the NASA Ames Research Center, we also ran our 1G control on the ground. So this is the 1G control on the ground that's essentially giving us our 1 gravity comparison with the experiment in space. So what did we do afterwards? We did 2 things. First of all, we took the liquid out and we examined biological leaching, looking at biomining, using inductively coupled plasma mass spectrometry to look at the elements in the liquid to see how the elements had been released from the rock and how much mining we had done. We also studied the growth of biofilms over the surface of the rock to study how the microbes grew. And this data is just being written up for a paper at the moment. So the data analysis is not complete. So today I'm going to focus on biological mining and not so much on biofilms. But if you're interested to know about biofilms, that data will be forthcoming. So today I'm going to talk about one particular group of elements that we studied, leaching from the basalt, and that is the rare earth elements. Now, some of you may be familiar with rare earth elements. They're everywhere around us. They are used in high technology. Your mobile phone is full of rare earth elements. If you have an electric car, the magnets in there are full of rare earth elements like neodymium. These are ubiquitous in high technology and electronic devices. And there are a range of them that I've listed here. I'm not going to go through these, but this is just to show you the huge diversity of uses of rare earth elements, everything from high power magnets, camera lenses, lasers, fiber optics, you name it. And there's rare earth elements in there. So these are crucial for high technology. Interestingly, they're not actually rare on the Earth, despite their name. So we're not going to go into space and mine these things to bring them back to the Earth. That's the first thing I want to point out. Whenever you talk about space mining, people think you're going to go off into space, collect these elements and bring them back to the Earth. But I want to point out that rare earth elements are not economically viable to mine in space and bring them back to the Earth. But if we're going to build permanent settlements beyond the Earth, on the Moon or Mars, we may want to mine these elements locally to support manufacturing processes beyond the Earth. So these are the data, and I want to just show you some of the data that we got. This is a paper, if you're interested, that we published this data in at the end of last year, looking at this rare earth element extraction. These are some data. This is a particularly useless slide because you can't really see these data. So let me move on to the next slide that shows you some of these data. And what you're looking at here are the space experiments in the upper slide and the ground one gravity experiment in the lower slide. On the x-axis, you can see the different rare earth elements. And on the y-axis, the percentage increase in leaching of the elements compared to the control. And what I want you to see here is that Sphingomonas desicarbalis, one of our microbes, managed to increase rare earth element leaching from the basalt by between about 160 and over 400% compared to the non-biological control. And you'll also notice, which is interesting, is that it tended to increase the leaching of heavier rare earth elements on the right-hand side here compared to the light rare earth elements on the left-hand side. And you'll also see that it did the same thing on the ground, which you can see in the bottom slide here. Two of our organisms did not biologically mine rare earth elements. They did mine other elements, but I'm just focusing on rare earth elements in this slide. Interestingly, I just put this up here on the slide. Could we also think about rare earth element leaching as a biosignature for life on places like Mars? In other words, because the biology has this preferential effect on heavier rare earth elements versus light rare earth elements, might this also be a biosignature? Now, this is triplicates. I've not put the error bars in. I'm sure hopefully some of you are thinking, where are your error bars? The reason why, if I put error bars in all these curves, you just end up with a big vertical line. But have no fear, these are the statistics. And what I want to simply point out is that the biological experiments caused a significant increase in leaching in the Mars and the Earth gravity conditions. One of the things that we noticed was that the microgravity was not significant. But one of the reasons why this was the case probably is because we lost one of our microgravity control experiments. And as a result, the standard deviation in the control was greater than the standard deviation in the Mars and Earth gravity controls. But interestingly, the mean leaching in the microgravity was similar to the leaching under Martian and Earth gravity conditions. So we think that we lost statistical significance because we lost one of our controls, but the absolute quantities of leaching were much the same as in the Mars and Earth gravity control. Now, remember I mentioned that hypothesis about there would be different effects of gravity because of the different effects of fluid dynamics. Well, in fact, in this experiment, we did not find any significant difference between the gravity conditions and the leaching of the elements between gravity conditions. And that was interesting because we expected there would be, because of this effect of gravity on fluid dynamics, we expected there to be a difference in the total quantity of elements leached from the rocks in different gravity conditions. So why was gravity not significant? Why did we see this unexpected result? Well, another thing that we did in this experiment was to look at the total cell numbers at the end of the experiment. And this is rather complex data. I'm not going to go through this in detail, but all it's showing is the cell concentrations in the different gravity conditions for the 3 different organisms, both with respect to direct cell counting and optical density. And the simple result that comes out of this quite complex set of data is there was no effect of gravity on the final cell numbers. And this might be the explanation for the reason why there was no effect on the mining of elements, was because the total cell numbers in all the different gravity conditions were also similar. In other words, the similar cell numbers led to a similar level of leaching in the gravity conditions. But then you might ask yourself, but why were the cell numbers the same given the different gravity conditions? And we actually don't know the answer to that. And one hypothesis we have is that because our experiment was much longer than most microbiology experiments on ISS, our experiment was 20 days long because we wanted to look at the leaching from the rocks. It's possible that despite the different gravity conditions, the cells were able to reach the same final cell number in the experiment regardless of gravity conditions. And this raises the question as to whether in the early phase of the experiment, perhaps the growth rates were different in the different gravity conditions. And if you look at other people's data, what you'll see is that in different gravity conditions, cells tend to grow at different rates because of the lack of mixing and the lack of sedimentation. And what we think is that maybe our cells grew differently in different gravity conditions at the beginning of the experiment, but once they reached stationary phase, once they reached the final cell number, then there was no difference between the gravity conditions. So of course, to test this hypothesis, what we would really need to do is go back into space and measure the full growth curve of the cells during the experiment, which we were not able to do in BioRock. But of course, that could be something we could do in the future. But what's interesting is it shows you that in principle, you could do biomining on asteroids and Mars. And if you leave the bioreactor for long enough, the biological mining might be identical in the different growth conditions. Another question that we don't have an answer to, and we're going to be looking at this summer in more detail, is how did Sphingomonas desiccabilis do the leaching of the rare earth elements? Well, a very common mechanism for cells to leach elements from rocks is by acidification. Some microbes make the pH go very low, they acidify the medium, and as a consequence, the rocks are essentially made acid and they release the elements. This is the basis of biomining of copper and gold, for example, the acidification of the rocks. In our experiment, the pH actually goes slightly alkaline rather than acid. On the right-hand side here, you can see a ground experiment where for the first 21 days, you can see the control is essentially at pH 7, and the 3 organisms go slightly alkaline. Then there's this very big drop when we add the fixative, which acidifies the medium. But during the main phase of the experiment, when the leaching is occurring, it actually goes slightly alkaline. So we think that pH is not the reason for bioleaching. Our working hypothesis is that Sphingomonas desiccabilis is leaching by producing extracellular polysaccharides, extracellular polymeric substances that are sugars, essentially long-chain sugars that bind to rare earth elements and sequester rare earth elements from the rocks, from the medium. And this is well known. It's well known that EPS can bind metals, and that's because it has groups like phosphate groups and sulfate groups that have negative charges and can bind metal ions. Interestingly, there was an experiment was carried out by this group to study rare earth elements in natural biofilms in streams. And they showed that biofilms with copious quantities of extracellular polymeric substances not only bound rare earth elements, but preferentially bound heavier rare earth elements compared to light rare earth elements, just as we saw in our own experiment. So this summer, we're going to be doing some experiments to study extracellular polysaccharides and to see whether they can bind rare earth elements, to try and understand the mechanism by which these organisms carry out rare earth element mining. So this, of course, is interesting because it shows you how space exploration can advance Earth-based problems. If we want to do environmentally friendly rare earth element mining on the Earth without environmental destruction, maybe the experiments we do in space can motivate us to understand the mechanisms of rare earth element binding and improve our ability to carry out these experiments on the Earth. I should also say something about the yield of our experiments. In commercial biomining operations, extraction of elements ranges from about 8 times 10 to the -3 to tens of percent. We are at the lower end of any commercial operation, but remember that this is an experiment, not an optimized commercial operation. We had other objectives in BioRock. For example, we wanted to study biofilm growth across a contiguous rock surface, so we didn't crush our rocks. And in a commercial operation, of course, you would crush the rocks to present them to the microbes with a greater surface area. So this was not an optimized biomining setup, so we have quite low yields. But of course, this could be improved if you're really trying to build a commercial biomining operation beyond the Earth. But nevertheless, what was exciting about this is it was the first biomining experiment in space and the first demonstration of mining and biomining under altered gravity regimes. So just thinking about this now, just standing back and thinking about applying this to places beyond the Earth, where would we go and get rare earth elements in space if we were building permanent human settlements beyond the Earth? Well, one place we can go is the Moon. And intriguingly, some of the rocks on the Moon have enriched rare earth element concentrations. So the Oceanus Procellarum, which is this big sort of heart-shaped basalt region in the middle of the Moon. When you look up at the night sky, you can see the Oceanus Procellarum in the middle of the Moon, and it's made of creep rocks. And creep is an acronym for potassium, rare earth elements, and phosphorus. And it's enriched in these elements partly because of the way in which the Moon differentiated when it first formed and the way in which these incompatible elements were sequestered in the basalt. These rare earth element concentrations are about 10 times higher concentrations than the rare earth elements in typical basalt, which we used in our ISS experiment, but they're still about 10 to 100 times less than rare earth elements in ores on the Earth. So that's why I point out that at the moment, it's not economically viable to go to the Moon and mine rare earth elements to bring them back to the Earth, because we have plenty of rare earth elements on the Earth. We don't need to be going to the Moon. But if you were on the Moon building a permanent settlement and you wanted rare earth elements for electronics, for example, you might try and get them out of creep rocks, because that may well be more economically viable than digging them off the Earth and taking them up to the Moon. Having said that, there might be higher concentrations at sub-kilometer scales. So maybe there's concentrated areas of rare earth elements. Interestingly, the Chinese sample return mission landed in the Oceanus Procellarum in these regions of creep rock. So I'm really intrigued to know what they find about rare earth elements on the Moon. And then there may be rare earth elements on Mars. We actually don't really know about ores on Mars. We do know that Mars has giant impact basins and giant volcanic regions that are now extinct that once hosted hydrothermal systems. And hydrothermal systems are locations of most of our economic ores on the Earth, because hot fluids dissolve rocks, they concentrate metals that then precipitate and form economically important ores. So in principle, there should be economically useful ores on Mars. The problem with Mars is, of course, you can't take microbes there because planetary protection regulations tell you that you're not allowed to contaminate Mars with microbes. So in fact, you can't do biomining on Mars, but you could do biomining on the Moon. And this is an example of the way in which if you want to go out and do mining on other planets, you might use different types of mining in different places. So for example, on the Moon, we might do biomining in the sort of way that I've illustrated here. On Mars, we might have to resort to old-fashioned chemical processes. for example. There's a lot of course, interesting technical questions here, and a lot of ethical questions as well. You know, should we be mining other planets? Should we be extracting resources, and who owns those resources? Well, these are questions I don't have time to address here, but they're very interesting ethical and social questions. This is just a slide to show you the concentrations of rare earth elements in the biorock, basalt rock, that we sent to the International Space Station. These are typical concentrations in lunar creep rocks, so you can see they're about 10 times higher. And these are concentrations of rare earth elements in ores in China, like the Berea and Obo ores, and you can see that they're much higher concentrations than creep rocks. But nevertheless, creep rocks are still of interest on the moon, and we might want to mine these for rare earth elements. I also wanted to point out that we did a study of the extraction of other elements from rocks. Vanadium is a fascinating element. If you add vanadium to steel, you get high-strength steel alloys. So if you buy some spanners to mend your car, for example, a lot of those spanners that you can buy, high-strength steel spanners and other tools, have vanadium in them. Vanadium also makes steel very abrasion and thermal fluctuation resistant, which is of relevance to extraterrestrial environments. So in general, vanadium is a very interesting element from the point of view of extraterrestrial settlement. It also has very good neutron absorption capabilities. Vanadium is added to reactor materials in nuclear fission reactors to absorb neutrons. So it has applications in the nuclear industry as well. So if we want to build a permanent settlement beyond the Earth, we might actually want to get hold of vanadium. We can also use it in batteries and a variety of other things. And we've also published this. This says in prep, but it's actually now published. I should update this slide. It came out a week ago. So if you're interested, you can look that up. It's in Frontiers in Microbiology. And that's the vanadium results on the right-hand side. Again, not very useful. It's too detailed, but just to show you, we have some data, and that's vanadium biomining. So this is just an illustration of another type of element that we can extract from rocks using microorganisms. And in this case, both the Sphingomonas and the Bacillus subtilis was able to do vanadium biomining. Finally, I wanted just to mention BioAsteroid that flew at the end of last year. This was a flight on SpaceX Commercial Resupply Mission 21. It was the first commercial mining experiment that went into space. And in this experiment, instead of looking at basalt rock, we analyzed asteroid material. So we actually sent up pieces of meteorite into space to study the extraction of platinum group elements, things like iridium and platinum from asteroid material, and using both Sphingomonas and also a fungus. And then we also used a mixture of bacteria and fungi to look at a natural community. And we investigated the leaching of elements from these rocks in space. So BioAsteroid was essentially biorock, but with meteoritic material and using fungus. It was a much smaller experiment. We only had one gravity condition, which was microgravity, no Earth gravity control, and no Martian gravity. But of course, we already have some results from the biorock experiment that we could compare to these experiments. So this is, I would say not as optimum as the biorock experiment, because we did not have a space one gravity control. So it's not as good, but we managed to fly it within 1 year instead of 9 years by going through this commercial route to launching our experiment. So these are some of the trade-offs in trying to organize space experiments. And then, of course, all of this work has been supported by facilities in the UK, the Science and Technologies Facilities Council, the UK Space Agency, the European Space Agency, Kaiser Italia, and Kaiser Space. And I'm very grateful to them for supporting this work. So I'm going to end there. I'm going to stop my screen, and thank you all very much for listening. I want to say again to Cardinal, thank you very much for inviting me to attend this fantastic conference. It's a real inspiration to take part in this. And I think maybe we have some time for questions.
Yes, thank you so much for participating. Your presentation was amazing. Now we have a lot of questions actually. First question from Öznur Köşker. Why is it needed to use double chambers on bioreactors? What is the specific purpose?
Yeah, that's a really good question. So the reason why we had two is just because we wanted to get as many experiments into a smaller space as possible. So in the cubic centrifuge, you've got this slot that you put your experimental apparatus into, and we can't change that. That's just the design of the incubator. So what we want to do is, given that we have a certain number of slots, we couldn't put 3 in because then there's not enough space for the medium and the fixative. And if you have 1, then you're wasting space. So it turns out that we were able to put 2 of those into our bioreactor, and you can see 2 of them. So this slots into the cubic centrifuge, and because we've got 2, we're essentially able to do 2 experiments in 1 piece of apparatus. So it's really a trade-off of trying to get as much experimental, to do as many experiments as possible in a smaller space. But as I say, the problem is you can't have more than 2 because you need to have your medium and your fixative chambers in there as well. So, it's just it's a trade-off of doing the experiment, but having as many experimental apparatus as possible. It's a good question, though.
Thank you. Our second question from Ulash Barut.
Yeah.
What do you think about a molecular biology and genetics students should do to work in astrobiology while in college? I'm also very excited.
I think that's an excellent question. So just to give you a bit of perspective from my own background, I did my PhD in medical molecular biology. I actually studied blood clotting proteins because astrobiology did not exist. And my advice to anyone who wants to get into astrobiology is don't become too obsessed and focused on being an astrobiologist, because many of us are not actually astrobiologists. So people who study exoplanets tend to be astronomers. They come into astrobiology. People who study life in extreme environments tend to do a degree in microbiology or molecular biology, come into astrobiology. In fact, it sounds a bit strange, but when students ask me, how do I become an astrobiologist? I often say to them, don't. Focus on one area of science and develop a good depth of knowledge in one area of science, and then bring that into astrobiology. So I think if you're doing molecular biology or genetics, that's a really good thing to be doing, because then you come into astrobiology with a very good expertise in one area of science. So the way in which you can use that expertise is to go along to astrobiology conferences, make connections with other astrobiologists, and continue discussions, come along to conferences like this. And then eventually, later on in your career, you can do astrobiology more permanently. I mean, I should say, so I talked to you today about biorock, but many of the things I do in my lab are actually not astrobiology. I have funding, for example, to study weathering in microbes and the way in which microbes break down rocks, for example. So there's a lot of non-astrobiology work that we do in my laboratory as well. So I think you'll find this generally. So that's a very long answer to your question. But basically, I'm saying, don't worry about having to be an astrobiologist. Just continue to take an interest in astrobiology, get involved with it, and use your knowledge of molecular biology and genetics to contribute to astrobiology questions.
Thank you so much. Our next question from Melike Naz Şayan. Sir, I know that on Salda Lake in Turkey, there's a similar mineral formation in Jezero crater on Mars. Can microorganisms who live in Salda Lake give us information about possibility of life in Mars?
Yeah, that's also a very nice question. Of course, we don't know whether there was any life on Mars, but we can use these environments that are similar to Mars to understand some of the physical and chemical extremes that limit the possibility of life. Now, of course, you have to realize that the Earth today is very different from Mars, even in its past. Today, the Earth has a lot of oxygen in the atmosphere. And so when we go to a lake on the surface of the Earth today, it's full of all sorts of microbes that can use oxygen and the rich organic material that comes from photosynthesis. So, of course, even on the early Earth, even on early Mars, rather, we would not have expected lakes to be like the lakes we see on present-day Earth. But nevertheless, we can learn things about the limits of life on Mars and the limits of life, and whether we might be able to try and look for life in some of these extreme environments on Mars by studying lakes like Solda Lake in Turkey. So absolutely, studying the extremes of life in these lakes, like the lakes in Turkey, absolutely gives us really important information for thinking about how we might explore lakes on Mars and what sort of conditions may or may not have supported life there.
Thank you. This question from Emre Vural. Do you think we are likely to encounter silicon-based life forms in the universe instead of carbon?
Yeah, that's also a very interesting question. I mean, my own personal view, and I think this is not so much a personal view, it's based on chemical view, and I think a lot of people would agree with this, is probably not, because when silicon comes in contact with oxygen, it tends to form rocks. And in many ways, if you look out of your window, you will see silicon-based chemistry on the Earth, and it's called rocks. And there are a vast number of different minerals on the Earth. So some people say, well, imagine a planet where there's lots of strange silicon chemistry going on. And what people forget is that there has been 4.5 billion years of silicon chemistry happening on the Earth. It's just that when silicon binds to oxygen, it tends to form these inert, long-chain silicon compounds, rather than the complexity of compounds we see with carbon. Another really interesting thing that I find interesting is when you look at what biology does with silicon, it does rock-like things with it. So, for example, sponges form these structures called spicules, which are these support structures that hold up sponges. Plants have things called phytoliths, and phytoliths are essentially silica structures that provide structural support in plants. Some plants can have 7% silica in their structure. And so what's interesting is when biology gets hold of silicon on the Earth, it does rock-like things with it. So I suspect that there is always going to be silicon in life, but we'll never see a life form that's completely constructed from silicon. But never say never, because if you have a planet with very low concentrations of oxygen, you can get more interesting silicon chemistry that isn't just long chains, but actually you get these cage structures. So maybe on a planet where there's less oxygen, there's a chance for little pockets of silicon compounds to not form rocks, but some sort of fluid. Maybe you might have some sort of replicating silicon-based life form. So I think that's a very interesting question. I think the answer is still open. My general view is I think carbon is a more likely backbone and fundamental element for life rather than silicon, but I wouldn't be so narrow-minded as to say it could never happen.
Thank you so much. That was a great explanation. Melike Nasrayan asked, which extreme environments is used more in research of life in Milky Way galaxy?
Yeah, that's also an excellent question. Of course, we are limited to understanding life in the rest of the Milky Way galaxy because we only have one planet that we currently know that has life, and that is Earth. So the extreme environments we use to assess life throughout the rest of the galaxy is necessarily limited by extreme environments here on the Earth. And of course, there's a whole diversity of extreme environments on the Earth, from the lakes in Turkey that we heard about earlier in someone's question, and then we think about the polar regions or life deep underground. There's a very fascinating variety of extreme environments on the Earth, but nevertheless, they are still all Earth environments. So although we have lots of biological material, we still only have one planet to assess or to do research on life in the rest of the Milky Way galaxy. So we're still very limited in our biological understanding of what might be possible beyond the Earth. So that's a very nice question. Thank you.
Thank you for answering. Another question from Emre Vural. Could tardigrades floating freely and dropped in space evolve elsewhere and sustain life?
Yeah, that's also an excellent question. So there's two problems in your question. First of all, could tardigrades drift through space and survive? And of course, a tardigrade is a very robust and very extreme tolerant creature, but it will accumulate radiation damage if it's just drifting through space in a dried, desiccated, vitrified state or whatever state it's in. And eventually that radiation will kill the tardigrade. So the first problem the tardigrade has is it mustn't be drifting around in space for too long. It has to find a planet to land on. And then, of course, the question is, as you rightly point out, once it drops out from space, can it evolve and be sustained? Well, tardigrades require food to live off. They require algae to graze on, and they also respire using oxygen. So they have to land in an environment where there is food to eat, probably existing microorganisms, and also some oxygen. So although tardigrades are very extreme tolerant, and you can dry them and you can expose them to radiation, they're still, like all animals, a little bit fussy. They need particular types of food, and they need particular radiation conditions and particular gaseous conditions. So if they land on another planet, they're going to need lots of liquid water and food if they're going to be able to evolve and sustain life. So they sort of need somewhere like Earth, really.
Thank you so much. Simaki asked, did you realize any mutation in microorganisms after the experiment?
Again, another excellent question. We are sequencing our microbes, but we don't think that there'll be mutations for the simple reason that our experiment is actually quite short. I mean, the experiment is long compared to most microbiology experiments. It was 3 weeks long, but they weren't exposed to very intense radiation. Remember that the International Space Station is below the Van Allen belts, so it's quite protected. It's not in interplanetary space. So the radiation environment on the space station is higher than the Earth, but it's still not very, very high. So we may not have particularly significant radiation effects in our microbes, but we don't know. And we have the microbes, so we might as well do sequencing on them and see what happens.
Could we see mutations if they were longer, they stayed there longer?
I think so. I think it would be easier if you had them there for longer. It would be nice to expose them to, the interplanetary environment or the radiation environment on ISS for, a long period of time and see what happens.
Why didn't you leave them there for longer?
Oh, that's a good question. The reason is that they had to come back on the Dragon capsule that we sent them up to space station on. So we had a limited time period for the experiment. They went up there on Dragon and they came back on Dragon.
Yeah, I understand. Thank you. Emine Gergen asks, some work has been done on artificial gravity and people have been rotated around a giant centrifuge. How healthy is the use of this fast-spinning centrifuge? What is the negative impact?
Yeah, so you can, of course, you're talking about increased gravity using a centrifuge. That's also an interesting area of research, not just microgravity, but increased gravity. I mean, what happens if you have a life form on an exoplanet that's 10 times Earth gravity? So we actually don't know much about increasing gravity on other planets. And how healthy is the use of a fast-spinning centrifuge? Well, of course, this increased gravity in a centrifuge is not just increased gravity, it's also Coriolis acceleration and other factors. So a centrifuge doesn't just increase gravity. As you sort of rightly point out, there are other effects on biology as well. And we actually don't really know what the negative impact would be on, say, microbes living in hypergravity for long periods of time. So that's an interesting question.
Thank you. Özgür Koşker asks, measuring full growth curve is needed, you said. How can you proceed after obtaining the growth curve? How you can determine where to obtain maximum yield of rare elements?
Yeah, so what would be nice to do is go back and do that experiment, but this time have some sort of turbidity sensor inside the container, so we could measure the turbidity or the growth of the microbes in the container. Then we could get the whole growth curve and we could see whether gravity affected the growth early on in the experiment. So that's something we would like to do. It just needs a technical development of the apparatus. How can you determine where to obtain maximum yield of rare earth elements? That's also a nice question. Of course, we were doing an experiment, not a full demonstration of biomining. I think one would need to do experiments where you crushed up the rocks. We could also go and improve our understanding of the rare earth elements on the moon, for example. Where would be the best place to get the maximum yield of rare earth elements? So there's a lot of unanswered questions when you come to thinking about doing this experiment on a commercial scale. And there's a long way to go between the experiment and a true sort of commercial bioreactor on another planet. But I think these are all very interesting questions, and hopefully we can do this experiment on the moon one day.
Hopefully. Nitya Palekar asked, "Hello, sir. What were the other challenges apart from the many samples involved that you faced while planning out this experiment?"
Yeah, thanks for that question, Nitya. Doing space experiments is challenging, and I had to learn this from scratch. And the difficulty is that when you go into a laboratory on the Earth, you can just go in and do your experiment, and if it doesn't work, you can come back and do it again. If I wanted to do the BioRock experiment in my lab here in Edinburgh, I could do this in like 3 weeks, and we do these experiments in 3 weeks, and yet this experiment took 9 years to organize. And the reason is that you've got to build an apparatus that fits into the centrifuge. You've got to test this experiment several times over to make sure it's going to work, because what you don't want to do is launch it into space after 9 years and then it doesn't work, and then you've got to wait for a few more years to do it again, or alternatively no one wants to do it again because it failed the first time. So there's a lot at stake. You've got to make sure it's going to work. And then the other thing you have to do is make sure it's safe for astronauts. So this looks like a very simple little cube, but it has to be designed to be able to survive a rocket launch. It then has to be put in an incubator safely. It then has to have all the fixative inject automatically. It has to measure the temperature and microgravity, and then it has to survive coming back in a Dragon capsule. All these things take time, and they take a lot of interaction with engineers. And when you're a scientist like me and you don't know anything about engineering, that's a challenge, communicating with engineers and trying to tell them what it is that you want to do and have them design something that works for your experiment. And then there are administrative challenges, getting a rocket flight, getting on the schedule for a rocket flight, preparing the experiment before the flight, handing it over for launch. All of these things take time. So it's a complicated thing. I would say that it's getting a lot easier. So the bioasteroid experiment we organized and flew in 1 year. And of course, we had the experimental apparatus already, so we didn't have to redesign this. But quite apart from that, it's also just getting easier to fly experiments to space station through commercial routes. But it's a complicated thing for sure. And, it's something that we don't do all the time because it took 9 years. This was a project that was run alongside lots of other projects we had going in the lab. So this is something that I wouldn't recommend doing full time, just because the opportunities are very few and far between.
Thank you. Hashim Aktaş asked, what would you recommend astronomy clubs to follow so they can contribute to astrobiology researches as an amateur?
Yeah, that's a really good question. Of course, I'm a microbiologist, and it's difficult to do amateur microbiology because you need a microbiology lab. But there are other things. For example, there are citizen science projects where you can look at exoplanet data and try and find new exoplanets. There may also be trips you could do to extreme lakes to look at life in lakes and to explore the environment around a lake. So we heard about the Sabka lakes in Turkey. If you're talking about a Turkish astronomy society, maybe you can go to these lakes and you could organize a field trip to study these ancient lakes and walk around them and think about Jezero crater, maybe take some maps of Jezero crater and see how it's similar to these lakes. So I think what's exciting about astrobiology is you can go to some quite interesting field sites. And in Turkey, you have fantastic field sites with different lakes and other sorts of extreme environments that you can visit. We don't have so many in the UK. I mean, Scotland is sort of extreme in the winter, but it's not really Mars. But you have fascinating salt lakes that you can go to. So I think you've actually got a lot of exciting opportunities for astronomy clubs to go and look at biology. And you don't need to be a microbiologist. You don't need to collect samples from these lakes and analyze them. But you could take a microscope. You can buy quite cheap amateur microscopes that you can plug into your laptop. So you could buy one of those and you could go to the lake and you could look at the microbes in the lake. You could think about the levels of salt. You can also buy quite cheap instruments that will allow you to measure the salt, or you could take some pH papers. and study the pH of salt lakes over time, maybe do a seasonal study or a study of the microbes under a microscope over a seasonal time period and study the way in which life changes. There's all sorts of great stuff you can probably dream up for an astronomy club to actually do an astrobiology project in an interesting environment.
Thank you for your answer. Our last question is from Emre Vural again. What do you think of similarity of the Laniakea, super piggy, and the brain?
Okay, so you're talking about our supercluster, I think, the galactic supercluster. Yeah, you know, it's funny how the human brain sees patterns in things. There's a really interesting paper, and I forget who it's by now. If you're interested, you can look it up. But these, it's looking at the structures in cells. So there's a structure in cell called the Golgi apparatus that's involved in processing proteins, and it's got a very similar three-dimensional structure to neutron stars. Okay, and there's a really interesting paper asking the question, is there a link, not that cells have come from neutron stars, but is there some sort of energy minimizing effect going on here, where biology is trying to minimize energy, so it ends up with this three-dimensional structure that looks a little bit like a three-dimensional structure in a neutron star that is minimizing energy. And they're like these parking ramp structures. And this paper is basically making this comparison. So you ask this question, is the Laniakea, super piggy similar to the brain? I think in that case, probably there isn't much similarity, but who am I to say? But I do think you raise a really interesting question, which is, are there similarities in biology and astrophysics that are actually linked by things like energy minimization? So we end up with structures in both in biology and at astrophysical scales that look similar because there's similar physics and similar laws of physics, like the second law of thermodynamics, playing out at the microbial, at the biological scale, and at the galactic scale as well. So it's an excellent question.
Thank you so much for your answers, and thank you so much for participating. It was an amazing presentation, and your answers were amazing. Thank you so much.
Thank you. Thanks for inviting me again, Carla. And all I just wanted to say to everyone is keep astrobiology going in Turkey. This is really, really exciting. I hope you form a full-scale Turkish astrobiology society and continue these conferences. This is a really, really excellent conference, and I'm very privileged to be asked to take part in it. So thank you.
Thank you so much. Goodbye.
Have a good rest of the conference. Bye.
Thank you. Our conference will continue after 7 minutes.
Hello everyone, I'm Zeynep again. We have met today, but for recently incomings, I will introduce myself first. I'm a member of Agenastro team of Molecular Biology and Genetics Turkey social media platform, and also I am studying bachelor degree of Molecular Biology and Genetics at Üsküdar University. And also I am currently a Chief research assistant at Medipol University. I will be hosting this session for you. I will briefly introduce Dr. Lev Horodyskyj, who will share a presentation about gamification and role-playing stimulation for astronauts and astrobiology today. He spent 8 years by working on the digital science curriculum at Arizona State University after completing his PhD in earth science and astrobiology at the Pennsylvania State University in 2009. His primary research interest in intercultural science education. That's why he is both an astrobiologist and an educator innovator. He is currently a visiting assistant professor at the University of Virgin Islands, and he is now with us. Hi, Doctor. Welcome.
Hi, thank you. Can you hear me?
Yeah, we can hear you. How are you?
I'm good. How are you?
Fine. I'm also fine. A little bit excited, but it's okay. Today it's last session for our conference, and now it's your turn. Would you like to add something more about you, or if you want, you can start your presentation. It's your choice.
I think I'll start the presentation because it covers a lot of what I've worked on and how I've come to think the way I do about astrobiology education.
Yeah, okay. Thank you. You may start.
All right, let me share my screen. All right, can you see my presentation?
No, not now. I cannot see. Did you click share button in the?
Oh, hold on. Select window. There we go.
Okay.
There we go. Is this better?
Yeah, now we can see. Okay.
All right.
The floor is yours. I'm going.
All right, excellent. So thank you so much for the invitation. So I'm going to be talking about gamification and role-playing simulations. A lot of the work I've done has been in astrobiology, but I'll talk more broadly about science teaching and learning because I believe it's important, especially if you're developing a new astrobiology program, to think about how we teach science to students to help them become scientific thinkers. It might get a little loud here for a few seconds. The Virgin Islands are in a bit of a drought, and so there's water being delivered to the neighborhood, and the truck is quite loud. So, all right. All right, so I'll go ahead and get started, and hopefully you'll be able to hear me over the noise. So I'll give you just a little bit of background on who I am. So as Zeynep mentioned, my background is I got a PhD at Pennsylvania State University in 2009 in geosciences and astrobiology. All right. And then I spent almost a decade at Arizona State University working on developing online astrobiology lab science courses. So my role there was as a lecturer, as an instructional designer, as a researcher, specializing in digital science education. Currently, I am building and developing a nonprofit called Science Voices, whose goal is to improve global science education around the world. And I'm also on a global sabbatical at the moment. Usually I'm based out of Arizona, but currently I'm traveling around the world and working in a variety of universities to try to truly understand what challenges they face in their science education, both for the students and for the instructors. So I was in Indonesia about a year ago at this time before the COVID crisis struck. So I wasn't able to complete my time in Indonesia. Now I'm in the US Virgin Islands. I should be in Ukraine later this year, and then hopefully in Brazil next year. All right. So So let me tell you a little bit about my experience with with graduate school, where I did actual astrobiology lab research as opposed to the science education research I do now. So my background was in studying the history of terrestrial environments. So I was particularly interested in what was happening during the Cambrian, because we have a lot of fossil records here leading back into the Ordovician, maybe some cryptospores. This was the state of knowledge in 2008 when I defended, end of 2008 when I defended. So there were various biological indications and fossil indications, geology indications that something interesting was happening during this point in time. But what exactly it was a bit of an open question. So the kind of work I did was really focused on looking at ancient soils. So this was a soil I looked at from South Dakota. There's the parent material here, this heavily degraded saprolite material over here, and then the paleosol over here that's very thick, very clay rich. And if you thin section it and look at it under the microscope, it looks very much like a modern soil with clay flow patterns and severe degradation, covered by a sandstone eventually as this whole sequence was buried. So I spent a lot of time studying the sequence as well as a few in the surrounding area. And so kind of the main story that came out of this was that the only really interesting pattern that came out was this phosphorus depletion near the surface, which was coming from apatite, which is a phosphorus-rich mineral that's present in the metagabbro here in the base, and persists in the soil until it suddenly disappears here at the top, with some indications that there could have been something eating it. Phosphorus is a limiting nutrient on Earth's surface, and it has to be weathered out of the ground in order for it to be biologically available. So it seems like something was on the surface eating this phosphorus, leaving the calcium behind and creating these clay flow structures. So I was enamored with the idea of what may have been on the surface. And so I got into my head that the surface was covered in liverworts, lichens, and mosses. And I got to this from my geochemical evidence, but then also looking at some carbon isotopes and extracted carbon material. And this is the kind of story that I thought was there. This is what I thought was happening on the surface. This is the story I wanted to tell. And so that was the story I told. And then that resulted in a large number of problems in my at my defense, because it turns out that the evidence wasn't actually that sound. And what my geochemical what the observations that I had actually made in the lab that were robust actually stated was something like this that was a lot less interesting. There's definitely this signal for what appears to be mycorrhizal fungi in the soil, eating up all this phosphorus at the top of the soil and dissolving away these apatite minerals. But what and mycorrhizal fungi are typically these they have mutualistic relationships with plant roots. So the idea of mycorrhizal fungi existing in the absence of any plants seems a bit strange, but at the same time, the evidence I had for plants on the surface was not very strong. So this was what I was actually able to say. And so I was hammered pretty hard at my defense for going beyond what the evidence said. But my idea was, why did I think science worked this way? Because my idea of how science worked often came from from movies, from reading about the history of famous scientists, and the storyline that exists about scientists is that they're these lone geniuses who stand against the world and need to convince the world that their idea is right. But it turns out that's more acting like a lawyer where you have a case to prove. And science is more of a process of determining what is definitely untrue rather than proving what is definitively true, because the truth ultimately cannot be accessed by human means. And I'll get into what I mean by that in a little bit. So I had this misunderstanding of how science worked, and it persisted through undergraduate and then through the end of my graduate career. So my question to myself afterwards was, how did I get to my thesis defense without actually understanding how science worked? That it's not about proving your case, it's about making observations and understanding what the observations are telling you, and not finding the observations that let you tell the story that you want to tell. So that was one of the major lessons that came out of graduate school, and that's something that I've been mulling over for the 10 years since then. But research wasn't the only thing I was doing while at Penn State. I was also teaching an astrobiology course called Earth and Life. And this course was an introductory level course for students who were not going to be science majors, teaching them about the history of life on Earth. So we got to talk about a lot of different topics: planet formation, the history of Earth, evolution, extinctions, all sorts of topics. And when I started teaching this class, I had gone to a prestigious university for undergraduate myself, where all information was transmitted via lecture. And so when I started TAing for this class, there were a lot of in-class activities, a lot of worksheets and things to do in the classroom, which to me struck me as a bit juvenile, I thought, at the time, because that's what we did in high school and grade school. We did activities in the classroom. When you're an adult, when you are in college, you sit there and you listen to the lecture. And it turned out that these in-class activities that I wasn't a big fan of ended up being quite interesting. They generated an interesting reaction from students. So this was one of the activities that I did for the students, called Make a Critter, Kill a Critter. So one of the ideas that was quite prevalent in the early and mid 2000s here in the US, and I think it's a persistent problem in Turkey as well, if I remember some of these global surveys that have come out, is that acceptance of evolution is very low. And so teaching evolution can be difficult because there's this idea of perfect creation. So I gave the students an opportunity to make a perfect creation. So their homework assignment was to make a critter. So design, create, build, draw, make some kind of critter and talk about its adaptive abilities. So these were some of the critters that students presented. There were some that were quite interesting and not very Earth-like at all, like these floating balls of gas that kind of floated around the planet and photosynthesized. There were some cute animals from our students who were more artistic, who enjoyed the assignment because it allowed them to engage with the class in a manner that was more comfortable for them, because they weren't really science students, they were more art students or language students, and this allowed them an opportunity to bring those interests into a science classroom. And then we also got some pretty nasty parasites that I think were universally designed by the female students in the class. But after the students made these critters, in the actual lab assignment, I put them together into groups and told them to create an ecosystem with all these different organisms, determine who eats whom and what's going to happen over time if these patterns continue. And so what they found was that over time, their perfect animals needed to be modified based on how their animals interacted with each other. And then we started moving into a slow climate change, so a slow glaciation, which animals could move, which ones couldn't, and how would they adapt in response. A few animals went extinct, mostly because they decided to start on an island and they were not able to get off the island as the planet cooled. And then the last one we did was just an asteroid impact, so catastrophic climate change, to see who survived and who didn't. And this gave students a very good idea of what actually happens in evolution and how organisms in biology respond to each other and to changes in the geological environment over time. And this was something that students continued referencing throughout the entire semester. So this was a very early kind of form of role-playing, not exactly a simulation like you would think of a computer, but it was kind of a thought game, a role-playing game where they wanted to see who would win under all these various circumstances. Okay. So the lesson that I got out of doing activities like this was that games, art, and role-playing can help students retain ideas for longer, because several months later, they were still referencing this experience and using it to explain new concepts that we were encountering later in the class. Another assignment I had them do was to think a little bit more. I wanted them to think more deeply about the fossil record, a frequent creationist criticism back in the 2000s. I think it's become less prevalent now, was that, well, where are the missing links? We haven't captured every fossil possible, so therefore we can't prove that evolution happened because we haven't preserved every intermediate organism. And so what I tried to do with this experiment, with this assignment, was to get students thinking about how we can tell stories about what happened even if we don't have all the information. So for this assignment, I took pictures of myself every 30 minutes for the entire day, from the time I woke up to the time I went to sleep. I gave my students all of these photos, and then I asked them to reconstruct my day as accurately as possible using the photos and any additional resources they could find. A few people were able to find my website and my blog and get more detailed information, but for the most part, students stuck with the photos. And what was quite interesting about what the students constructed was that their reconstructions, although somewhat correct, revealed more about them than those reconstructions revealed about me. So for example, there was no picture of me eating breakfast, and in the assignment, one of the questions I asked my students was, did I eat breakfast? About half the class said, no, I did not eat breakfast, because there was no photo of me eating breakfast. And the other half of the class said, I did eat breakfast, but it was faster than a half hour. That's why there was no photo of it. And from looking at those reconstructions, I could understand immediately which students ate breakfast and which ones did not, because the ones who ate breakfast interpreted the lack of a picture of me eating breakfast not as evidence that I do not eat breakfast, but that I likely eat it just like they do, but it was shorter than the time frame in which the pictures were taking place. Whereas the students who did not eat breakfast did not see evidence of me eating breakfast and assumed that I also did not eat breakfast just like them. Okay, which was a very curious, result when I did this assignment. It turns out I did eat breakfast, and it's just the resolution wasn't good enough to catch it. But this happened a lot throughout the entire assignment. And for the first time, I started thinking about what are my students actually bringing into the classroom, that they're not just empty vessels waiting for me to fill their empty heads with knowledge. And so what I kind of discovered through this type of activity was that student experiences influenced how I interpreted what I showed them. And so that also went a long way in informing how I thought about education. And when I present ideas to students, recognizing the fact that I am presenting ideas into a very active mindscape, that different minds may react differently to what I am showing them. Okay, so these are some of the lessons from graduate school that helped inform the approach I took to teaching and the direction that I headed in after grad school. So some of the main lessons I got here was that based on my failures at my defense, was this assumption that there's this idea that knowledge about how the scientific process operates is assumed to have been taught somewhere else. So when you get to the college level, your professors think that you already learned how to do science and how to think scientifically somewhere else, in high school, in grade school, in playing and going to a museum. And it's not a concept. Scientific thinking is not a concept that needs to be explicitly taught. So everyone in colleges at the university level seems to have this assumption, but the reality is that coming up through a lot of these lower level educational systems, the primary and the secondary level, you're mostly learning science facts and not how those facts are actually discovered. So you're learning more about the results of science and not the process itself. But a lot of faculty have this assumption that surely you have been taught how to think scientifically and do scientific work elsewhere, so I don't need to reemphasize it. I can just teach you even more detailed content. And that becomes problematic for people like me who didn't really understand how science worked and get to our defense trying to prove a case instead of looking objectively at the observations we've made. Okay. And something else that was of interest was that student conceptions about how the world operates is influenced by their experiences, not just by how many facts they are exposed to. And this is something that's a big deal here in the US when it comes to climate change education, because a lot of scientists have the attitude, if I just tell people more facts, they will understand that climate change is a serious threat and will do something about it. And it turns out that it does not work that way at all. And I also started to discover that team activities and role-playing can be more engaging and effective than just lectures. So when I went to Arizona State University, where I did a sort of postdoc, it wasn't a true postdoc because I had a retirement plan, which postdocs typically don't have, I was able to start exploring some of these ideas in more detail, and particularly in a digital environment. So the main project I worked on during my time there was a course called Habitable Worlds. And this course was designed for undergraduate students to introduce them to astrobiology. So to teach them concepts across all of science. So these are just some of the screenshots of what we were working with. And I'll get into a little more detail of some of the philosophy that went into designing this course. So this course was organized around the Drake equation. So it was a half-semester course, which is what Arizona State University typically likes to do for online students, which means that it ran for 7.5 weeks. But because it ran twice as fast, there was twice as much work per week as opposed to a standard 15-week long course. So 7 weeks, 7.5 weeks, and then here in the Drake equation we have 7 terms. Basically we could teach a term a week. Turns out we didn't end up quite doing that, but this is an excellent framework for structuring content in an astrobiology course, because what we end up doing is in the first week we do some introduction to science and how science works, and those lessons were initially extremely unsatisfactory, which got me rethinking on how science actually works, and I'll show you what I've come up with in just a little bit. And then afterwards we could spend a week on stars, so stellar spectra, nuclear fusion, element production, brightness, luminosity, colors. So a lot of basic physics concepts and some nuclear physics concepts. The next week we worked on planets. So for planets we studied formation of planets, how to detect planets through radial velocity and transit methods, which lets us get a little more into colors and into gravitation and Kepler's third law. And then we got a little bit into the chemistry of planets and their atmospheres. We spent 2 weeks on studying Earth-like planets because obviously there are a lot of topics to cover there, from plate tectonics to the stability of water, to the greenhouse effect, planetary temperatures, and geology. So this was a hefty unit, which left less time to cover some of these later units, but we did explore a few topics here on phototrophy, various forms of respiration and biochemistry, and going a little bit through the fossil record and thinking about the energetics of civilizations and sustainability. So we spent more time developing this part of the course than this part of the course, but all the topics were organized around the big question of, are we alone? And telling students, how do we go about answering this question, because we don't know what the answer is. Okay, so organizing a course around big questions like this can make the topic more engaging for students, because they'll understand why they're studying this. I've recently, this year, I've been teaching physics here to biologists at the University of the Virgin Islands. And it can make it difficult when you're teaching basic physics, where you need to talk about kinematics and motion, heat and thermodynamics, electricity and magnetism, some quantum stuff, some relativity stuff, some particle physics. That's a harder narrative to weave for students on how are all these topics connected, and why am I, as a biology student, studying particle physics, because I really don't care about it. But astrobiology gives a very nice framework in which to teach actually a huge number of topics in science, from physics and chemistry to biology and sustainability. So the course also had a course project, which I designed and had someone else build that was better at programming than I am. And in this project, the students got 500 stars, 500 fake stars. They weren't real stars, because we didn't want students Googling the information to see what planets were around it. They were stars that we had created for this project. And we streamlined the data set, because we really wanted to avoid doing a research experience for undergraduates, because when a student is first learning a topic, like let's say interpreting spectra or interpreting a transit curve, real transit curves and real transit spectra are incredibly convoluted, incredibly complicated. And you end up confusing the students more than testing whether they understand the concept. So what we did was we simplified. It's a simplified star system where every planet was, where every solar system was oriented 90 degrees, so that they didn't have to worry about minimum mass for finding their planets. And we only focused on six atmospheric gases here for them to look for, so that they wouldn't be overwhelmed by what a real spectra would look like, but still had a science-like experience in hunting for habitable worlds. And because there were 500 stars here, this wasn't really cheatable, because the stars were randomized for each student. So acting as a final exam, what the students could do, they could not share answers. They cannot say, oh, the habitable world is around this planet. But what they could do was they can discuss with each other strategies and how to go about finding it, and check each other's work. And we would often see this in the discussion boards of can you check my calculations? I'm not sure about this. The program isn't validating what I'm doing. And other students would respond with, oh, you've got a plus sign here instead of a minus sign. And so it really became a miniature scientific experience where students couldn't share answers, but they could share strategies and check each other's work, which is what we wanted them to learn anyway. And the students received a huge sense of accomplishment, because looking at a sky like this with 500 stars, and this is one of 3 panels. This is the habitability panel. There was also a planet hunting panel and a star classification panel that was just as much work. And imagine doing 3 of those panels with 10 to 20 tasks on each of these panels times 500 stars. We gave them certain tools to help streamline this. It feels very overwhelming, but once students mastered the tools and were able to solve the simulation, they had an enormous sense of accomplishment and became extremely excited when they found a habitable world, and some of them would race to be the first ones to find the habitable world, of which there were about 10 in this simulation. Okay. In order to build this course, we also used an intelligent tutoring system, and it's an intelligent tutoring system that I would advertise a bit more, but they went out of business, so they are no longer around. But in the 10 years that they were around, they kind of acted like an interactive PowerPoint, and slightly more than answer some questions as you're watching a video. What I could do was I could drop these custom-made simulators onto the slide, and then the system would recognize what was happening in the simulator. It would know where this probe was. It would know how many observations they've made, how much data they've collected. It would know the average of the data, the standard deviation of the data. And I say data, but it's not really data because the data it's calculated from an equation. So it's kind of simulated data. But it knew all this information that it could pass on to the intelligent tutoring system, and then I could actually see what the students were doing and react to something more than you chose A, that's correct. I could actually look and say, I could catch people trying to cheat. One of the assignments here was they would tell me how many observations they would want to make, and they would say 10, and then we would check to make sure that they actually made 10 observations. Or some of them would try to cheat the system by just clicking 10 times, saying, I made 10 observations. And that was something that we can catch and say there's no standard deviation on your data collection, which means you collected all your 10 observations at the same point. Move the probe around, you didn't do the work. Okay. So these simulators were designed by me as well, and then I wired them into these intelligent tutoring system and was able to make these very interactive and very engaging experiences for students that were incredibly experiential. We had someone else on our team who designed virtual field trips. This was 360-degree photography where you can look around your environment. And then embedding it in the intelligent tutoring system, I was able to add questions on top of this virtual field trip to ask them to evaluate what they're looking at, depending on the colors, the structures, and then to classify these things. I often also used humor and pop culture references to engage students. Students in the US who are interested in astrobiology tend to also be big fans of science fiction. So there were a lot of science fiction references throughout the course. This particular reference is to a video game called Portal, where you shoot portals onto walls and solve physics puzzles in three-dimensional space. And the game was very popular about 10, 15 years ago. And generated a lot of memes that you may have seen on the internet, such as the cake is a lie, because the computer that was testing you was also trying to kill you. And to make the quizzes in the quizzes and exams in the course a little more interesting than a generic quiz or exam, I wrote a storyline of a hostile computer that's trying to kill you as you go through the course, and your job is to defeat this computer by answering the questions correctly. And it turned out to be pretty engaging for a lot of students. Some of the humor was very mean because some of my preferred humor is quite dark and sarcastic. It didn't appeal to all students, so you have to deploy this carefully depending on what's popular in your culture and in your society and what will resonate well with students. But for a lot of students, when the quiz gave them a very mean response, if they got a wrong answer, they would start laughing, and they would write in the reviews that the humor, like the really mean humor, helped break the tension and just made them start laughing, and they became less stressed when they were doing the quiz. So there are various ways that you can make these classes engaging. And so some of the work that I did while I was at Arizona State University was using intelligent tutoring systems, using humor and references to popular culture. In this case, I restricted references just to science fiction. So I pulled references from science fiction across the entire century. So there were references to Metropolis and some science fiction from like the 1910s and 1920s, just to help keep the course from getting dated over time, as references to science fiction that was popular in the 2010s are now 10, 15, 20 years old. And then we also did research on whether these simulations worked or not. So these are some of the results we got from our research studies. If you're interested in more details, we have a paper in Astrobiology in 2018 that gets into more detail about how the course is constructed and some of these statistics. And we found that students were performing fairly well. They were getting A's. A lot of people initially criticized the project of, are they getting A's just because they're clicking? If they click enough times, it'll let them go through. And a lot of online experiences are designed this way, that if you make three or five mistakes, it'll tell you what the right answer is and let you go on with a point penalty. The intelligence tutoring system we were working with didn't really have that option. So the approach I took to designing a lot of this material was, you're going to sit there until you do it right, which is what my parents made me do when I was a kid. Sit there until you do it right. This resulted in a lot of frustration from some students because they're very used to the idea of if I show you that I'm never going to understand this, can you just let me move on? And they were not used to this idea of, no, you have to get this right before you move on. But what it also did, by forcing them to stop and not progress past something they really didn't understand, it started to trigger a lot of conversations in the discussion boards that helped them understand better than they would have if we had just told them what the right answer was, assess a point penalty, and let them move on. This doesn't work for all students. Students who tend to, so the grades here are A through E. A is the best grade you can get. E is a failing grade. So this would be like 90% and higher, 80 to 90%, 70 to 80%, 60 to 70%, and this would be below 60%. The W stands for withdrawal. So those are students who dropped out of the class or did not complete the class and withdrew from the class. So all of these students here in these colored pie charts would engage with the course. Even our failing students, they would come back and try again and again and again and again, even if they didn't understand, and even if they tried 800 times, which we did see one time. A student trying 800 times on, might have been close to 900 times on one question, or one activity. It was a planet building activity. And we kind of wondered, why didn't you jump on the discussion board and ask a question? Whereas students who withdraw, they're not willing to ask questions. They're not willing to try. As soon as they encounter a barrier, they disengage immediately. Okay. So even with failing students, there's a way of engaging them. We just need to figure out why they're failing, where they're struggling, and engage them in those areas, which we can do with simulations. We also ran a local community college in the Arizona region, in the Phoenix region, adopted Hab Worlds as a complement to one of the astronomy courses they were already running, and found that once they started using Habitable Worlds, students performed better, that the grades shifted more in the positive direction. And this is from our project. In the project simulator, where they're hunting for habitable worlds, the students are given $50,000 in funding, and they can spend that funding to automate some of their calculations, as long as they can prove that they know what they're doing. So if they can do 10 calculations correctly, they can pay to have it automated, and then they never have to do that calculation again, which is why they're able to analyze 500 stars in just 7 weeks. But if you submit calculations for evaluation for automation and you get them wrong, you lose the money and you don't get a benefit. So there's a little bit of game theory in operation there of, do I try over and over and over again until I run out of money and have to do all my calculations by hand? Or do I strategize? Do I try more here and less over here? Or do I have someone review my calculations before I try to submit them? And what we found was, because one of the questions was, do these A's actually represent understanding, or do they represent persistence? And what we found was, when we looked at the pattern in spending of money in the Habitable Hunt simulation, students who got good grades also tended to optimize their spending. They would spend the bare minimum necessary to automate everything, whereas students who did more poorly were more likely to spend all their money, which indicated there was something there that they really didn't understand and potentially never understood because they ran out of money before they were able to demonstrate competency. Whereas our failing students either did not know this mechanism existed or did not engage with the project at all. So this wasn't the only place where we were implementing this. So I also worked with a political science course called Do You Want to Build a Nation, where we use simulators to help students think about issues like climate change, where we would give them these tiled worlds that were developed to have various energy, food, and material budgets. And I was also able to tie in some very, very basic climate change models into this massive spreadsheet. And what the students would then do, these are political science students who don't really spend a lot of time thinking about science. All of a sudden, they were going in, because they had this data available, this very rudimentary simulator available, while they were role-playing their diplomacy games, they actually referenced the data in their country a whole lot more than they were able to do otherwise. And so they were able to come up with policies that were more realistic, because those policies, they were able to run some policy simulations on their countries to understand exactly what the consequences of some of their decisions would be. And this is a game that was already very heavily role-playing based. Students role play as they get together in groups and role play as 9 different fantasy countries, each with a different ideology. And in this particular course, they're studying political ideology and philosophy of governance. And so what we've started doing through my association with this course is start to overlay the science on top of that role playing, and start overlaying digital simulators that can help make that role playing more powerful. Okay. So what we discovered through both of these projects was that intelligent tutoring systems can allow novel curricula that are just as effective as in-person instruction. And that's a difficult case to make for a lot of online education, and something that we've been especially dealing with during the pandemic, where a lot of people have had to switch over to digital technologies that they were unfamiliar with, and many got stuck using materials and infrastructure that isn't particularly great, where technically you are teaching online by giving a Zoom lecture and doing some multiple choice quizzes online, but that's something you would do in the classroom. And you have a supercomputer in your hands here, and you're using it to ask multiple choice questions and watch videos. We live in a world where we can do high-powered simulations of reality. Why aren't students engaging with that? So this is just a bit of a spaghetti plot. It's a little difficult to interpret. And there's a paper, we have a paper in the Journal of Learning Analytics that discusses this in more detail. But basically, to simplify what's being said here, is each dot represents a slide in our online course, in the Habitable Worlds course. And the closer the dots are together, the more students worked on them at the same time. So what this kind of reveals to us is that we see sequences here. The course starts here. The introduction of this course is very linear. And then we get into the star unit here, which is the second week of course. And it takes more of this odd shape here. So all of these lines here are our training activities where they're playing with simulators and answering questions and watching videos, and everything highlighted here in yellow are the quizzes. So there are 6 units, or there are 6 chapters in each unit, and each chapter has a quiz associated with it. So you can see here, blue is our passing students. In the first week where they had quizzes, these quizzes cluster very closely together. So it looks like the students are trying to do the quizzes all together, maybe even trying to do them without doing the training activities first because the quizzes were due. But then they kind of realize after this that that's not a winning strategy, that the quizzes test what we teach you in the training activities. So our passing students then switch to a strategy of do the training activity, then do the quiz. Training, quiz, training, quiz, training, quiz. And so you see each of these new units becomes more linear. And then the project here clusters really far away from all these activities, which means that as students are completing these quizzes, they're then immediately jumping into the project to complete the similar activities within the project. So this project does not associate with any activity here in particular. The students who failed take on a different strategy. So their intro is very linear, and then they get into this hub and spoke pattern where all their quizzes are clustered together, and then we kind of loop out to the training activities. So what this communicates to me is what a lot of the failing students tended to do was they would know that the quizzes were due that day. They would start working on the quizzes, and then they would go to the training activities to find the answer. But there were no answers to be found because the quizzes were often in the form of do these calculations or solve this problem or set up the simulator to meet these conditions. And so that is not an answer you can look up. That is a process you need to understand in order to be successful. And then what we also saw was that week after week after week, they continue taking the same strategy. The quizzes are due. I'm going to do the quizzes. I can't do the quizzes. Let me look up the answers. And they continued with this strategy pretty much the entire semester and didn't start working on the project until the very end of the term. Okay, so by looking at the analytics that came out of this system, what we were able to see quite a bit was this very different behavior in strategy of how students approach the course. And we can see this behavior as early as the 2nd week of the course. So if we are aware that this kind of pattern exists and we're working with a system that can detect it, this also means that we can potentially rectify it instead of letting these students fail. Unfortunately, a lot of these systems don't allow you to do these kinds of real-time analyses. So the best a lot of these systems can do is you can download this data after the semester is over, analyze the data, and see what happened. So this is interesting, but not actionable. And the reason I care about the fact that some of these students have these unsuccessful strategies is that in Arizona, most of our population is white. We have a strong Hispanic population, not as much of the other demographics as well. And what I'm plotting here is compared to their prevalence, the demographics of the class, are these categories of students overrepresented or underrepresented in passing and failing categories. Okay, so what we see here is for failing categories, which are the red and the gray, failing and withdrawal, our minority students, our Hispanic and Latino females, and Black and African American females and males are very much overrepresented in the failing category. So not only are we detecting not only are we able to detect failing behaviors or behaviors that lead to behavior, we can also see that the online class has inequitable outcomes. Now, this isn't much different from a lot of other science classes. The difference is that when you're using digital systems that are good, you can actually do something about this. Whereas in a paper and pencil class, you're not really actually able to do much about this because you don't get much insight into student behaviors and student answers. Okay. So the lesson that comes out of here is that unsuccessful behaviors and inequitable outcomes can be detected early and potentially rectified. Okay. If you're interested in pursuing a strategy like this within your own university, let me tell you one of the big problems you may potentially run into, which we ran into when we were building the first version of Habitable Worlds in 2011. When this idea is new and exciting, the university is willing to throw a lot of money at it, and you're able to get a lot of grants to fund the development of an experimental curriculum like this. For a university, anything that gets students more excited and more enrollments means more tuition dollars. And so they have good incentive to build, to fund the development of an online course that is this interactive and this engaging. And because there's the possibility of doing research, the scholarly community is also interested in giving grants to do this kind of work. However, we also know that technology ages over time, and some of the things that worked 5 years ago no longer work on the internet, so these things need to be constantly updated. And this is where you'll run into a problem if you want to take an approach of creating these kinds of very engaging astrobiology experiences in the university setting, is that you may be able to get a lot of upfront investment, but if you don't have a strategy for how to maintain it and update it as internet technologies change, whatever you spent a lot of money building is going to fall apart, which is the problem that we had. The university pocketed all the student tuition fees that they took for the class, and didn't pass any of these student fees into the course like you would in a traditional lab course. So there was no money to maintain the course after it was built, and it became very difficult to get grants because no one wants to give you a grant to fix up something that's old. They want to give you a grant to build something new. So if this is something that interests you and you want to take this approach, keep in mind you're going to need to treat this like a technology product, which is you're going to be able to get a lot of upfront investment to get this off the ground, but you're going to need to have a business plan to maintain this and upgrade it over time. Otherwise, it's going to fall apart, and you're just going to end up spending a lot of money rebuilding another version of this from scratch, which is eventually what we needed to do. Not only because it was aging very badly, but our technology provider went out of business. And so we finished rebuilding this course, I think in 2018, 2019, after I left. It took that long to rebuild it. And then a year later, the platform provider went out of business. So now the group at ASU needs to rebuild the whole thing again in another system. So these are some of the challenges you'll face if you move into this environment. So I've kind of become less enamored with these simulators and digital environments, mostly because they're very expensive to build. They can be difficult to maintain. They don't have a long lifetime, and they're inequitable. Not everyone has fast internet. I'm teaching in the Virgin Islands now. It is not unusual for my students to lose power. It's not unusual for me to lose power or internet in the middle of a lecture. And so anything that requires high computational power or an always-on internet connection is going to be unfair and inaccessible to a large portion of the world. So I'm a fan of this project that's also being developed at Arizona State University, the Solar Spell project, which is a computer attached to a solar panel that creates a local wireless signal. And I actually have one of the computers. It's called a Raspberry Pi. So it's a microcomputer. And the way this project works is that library content is preloaded onto the SD card on this computer. So videos, PDFs, and all sorts of other things for teaching in remote areas is preloaded on this. You hook it up to a battery, turn it on, it'll generate a wireless signal, and students' phones, and a lot of students have smartphones, even in fairly remote areas, they can connect to this device and get digital teaching and learning content even if they don't have an internet connection. So my interests have started moving into this kind of low power, low bandwidth environment for digital teaching and simulation teaching, mostly because I think this is what most students experience. Most students don't have a top of the line computer that could run the simulations that I developed. Okay. So the lesson here is that flashy simulators and high-tech solutions generate excitement, but are unsustainable and inequitable. Okay. And so one of the other ideas that came out of my time at ASU is a reconceptualization about how science works. We often use a whole lot of complicated terminology for science, like hypothesis theory, double-blind, 5 sigma, and we throw these terms even at introductory students as if they explain what science is. But science itself, it's kind of like teaching someone how to cook by talking about blanching and steam baths and things like that. Most people, their first experience with cooking is going to be putting some water on a stove and trying not to burn it. So when we're introducing science to students, we need to start thinking about how science works in an everyday context. And the example I like to use when I introduce students to this is imagine that you are in a car behind another car and you're both stopped at a red light. The light turns green and the car in front of you doesn't move. What would you do? And most American students would answer that I honk the horn. And I tell them, you just conducted the scientific process in a microsecond. You made an observation, green light, another observation, car isn't moving. You've assumed that the person isn't paying attention, and so you've run an experiment to test that assumption by honking the horn, and then you'll see whether they move or not. So if they move, then that assumption was correct, that they weren't paying attention. But if they don't move and maybe their hazards come on, maybe that indicates that your assumption was wrong and that the car is actually broken and you may want to get out and help. Most people just drive around in America. But that's how people implement scientific thinking in day-to-day life. So we should try to connect when we teach science to this kind of behavior. So thinking about the observations students make, you can't make, but you can't build a model, a computer model, a physical model, or any kind of model with just observations. You need to fill in the gaps with assumptions, and the assumptions are what you actually test in the scientific process. And over time, those assumptions are replaced by observations. And so I kind of redefined a hypothesis not as an educated guess, but an assumption-dominated model. And I redefined theory as an observation-dominated model. And that any scientific idea can slip between these two conditions, depending on how many observations and assumptions are in your model. And this was an interesting idea I started thinking about. And really the point here was thinking about how the scientific process can be described more simply and colloquially without using jargon, because jargon turns students off and it makes us sound really smart, but it doesn't help the students understand how scientific thinking actually works. Okay. So the lessons from my postdoc-ish times are, if you give students models and simulators to play with, can be incredibly powerful and engaging. Big data analytics allow incredible insights into student behavior, but its potential is mostly unrealized. Digital teaching resources are expensive and time-consuming to develop and can be too dependent on unstable or uninnovative education technology companies. And assumptions about what kinds of technology teachers and students have access to can result in inequitable outcomes. We all assume everyone's working with the high tech that we can afford at our expensive universities, but most students are not dealing with that level of technology. So I'm not sure how much time I have. I want to leave some time for questions. Can I have 5 minutes more to finish up?
Yes, sir, you have 5 minutes to finish, and then we will go to question part. Okay?
All right, excellent. So I'll go quickly through the lessons I'm pulling out of my global sabbatical. This is still in progress, so I haven't had time to really sit down and analyze all these results because I've been jumping from place to place for the past 2 years now. So I was teaching astronomy at a local community college where I started trying to reformat my class into assumptions and observations. So to get students really thinking about observations and bringing their own assumptions in. So instead of saying, this is plate tectonics, this is how it works, or this is gravity and this is how it works, or these are the moon phases and this is how it works, I asked them to go outside and tell me what you see. Tell me what you see each night, and then we will develop our explanations from what you personally observe. So I started testing this idea here to see if it was viable. And I found that it is feasible to redesign a science classroom to, instead of loading students with facts about how the world works, actually start engaging them in scientific thinking by asking them to think about the world around them and to describe it. And then I use those observations to build activities in the class that build off of those observations towards our consensus understanding. My time in Indonesia was cut short due to the pandemic. I was only able to teach there for about a month and a half, which translated to about 4 lessons. But what became very obvious while I was there is that even some of the most basic education technology just failed to work in that part of the world. There were tools, simple tools that I've been using for a decade that just didn't work in Indonesia because they were built 10 years ago. They were not optimized for smartphones, or there was no internet connection. And this tool, even though it didn't need the cloud to work, for some reason required it. And without access to the internet, nothing would load. Okay. So this is the reality of education for a huge portion of the world. And this year I've been teaching physics at the University of the Virgin Islands. And I've started rethinking about how we teach this. So this is an example of one of the slides I've used where we start off with the observations students make through what they see, what they feel with their skin, and what they hear. We derive the descriptive equations for that, and from there move to the big unifying ideas of physics instead of presenting energy and atoms as this is the way it is. So for example, most science classes start by introducing what atoms are because they're critically important to learning about physics and everything. I didn't introduce atoms until about a month ago because what I explained to my students was we don't need atoms to explain motion. It's helpful to know it, but we don't need them. We'll introduce these models when we need them, and then we'll understand the conditions that lead to the rise of these models. Okay, so physics I thought would be a bit challenging to do, mostly because it's so equation heavy, but I found that even in a physics class, you can start with personal observations and assumptions and then co-construct to the consensus scientific knowledge. Guide students to that understanding instead of telling them this is what the understanding is, memorize it. Okay, so far what I found is that science classes can be redesigned to demonstrate the scientific process in every lab, lecture, and assignment, and not just talk about it on the first day, which is what a lot of science classes do. You can actually engage students in scientific thinking every day of the semester and not just teach them facts. We also did a lot of work at home because we couldn't go in because of the pandemic. And so the students enjoyed the lab work where they could just use garbage and stuff around their house to understand how some of these processes work instead of spending a whole lot of time in a lab trying to operate complex equipment that they're never going to use again. And many, many students in classrooms have even fewer resources than you think. Okay, so I'm going to skip over because I think I've run way over time. My organization, Science Voices, that I'm running and building now, is going in a direction of helping address some of these problems. So you can go to the website to learn more about it. One of the things we're developing is an online education system that's simpler, that can integrate with instrumentation, so that we can do adaptive learning even if you don't have a fancy computer, or even if your teacher is technologically illiterate. So there are examples on the website of what this will look like if you're interested in either helping with the build or testing it or developing stuff in it once it exists. And then we have a number of people from around the world that are working on this. And then the other project we're working on, spinning off of that political science class, is the Greenworks project. What we do is we do a 5-week program where students around the world form teams, play this game, and then we fund them to do community environmental projects. Right now we're working with Indonesian students and Brazilian students. But otherwise, I'll just leave this here. If you want more information on what I'm working on now to help improve science education globally, especially when it comes to using digital technology, I've got a number of writings and ideas up at sciencevoices.org. And the platform that we're developing that I'm really excited about, we're going to be testing it here in the Virgin Islands over the summer, agavi-ed.org. And then I have my website with my portfolio at horodyskyj.net. Right. Okay.
Thank you for your awesome presentation, Dr. Lev. It was really remarkable and interesting topic for us. And we have so many questions now. By your leave, I will guide those questions to you from our audience.
All right. Excellent.
Our first question from Mal. I'm molecular biology major and I'm interested in gamification. I want to develop game simulations about evolution for middle high school. How did you find inspiration about your teaching techniques?
Okay, so for developing the simulators, the ones that I developed, that I enjoyed developing the most, were the ones that I had the most difficulty with conceptually. So plate tectonics is a difficult topic for people to understand, not specifically plate tectonics, but the inorganic carbon cycle. So this idea that carbon dioxide gets pulled out of the atmosphere through weathering, it ends up in the ocean as limestone and doesn't come back into the atmosphere until it's subducted and vaporized and comes back up through volcanism. So that's a concept that's good for gamification because it helps you visualize the flow of inorganic carbon through a system, and it's a topic that people don't really understand, the inorganic carbon cycle. Another concept I've had difficulty with is redox chemistry, especially when it comes to biochemical reactions. So I designed a simulator that helped illustrate that particular concept as well by letting you create all these molecular biologies. And you can see the electrons flying off of the food particles and forming waste particles and powering up the cell to do something and creating all these alien biologies. So I usually target concepts that I personally had difficulty with, where a moving animation or something you can poke would be useful to have. And if you're going to build these, treat them like a toy. A lot of people build these simulations as video games that you could only use them in one way. I always build mine as toys where there are lots of different knobs and levers that you can manipulate to get different combinations. It makes the simulator more usable outside of your immediate classroom.
Okay, thank you. Now our second question from Emre Vural. Are there any abiotic and evolutionary simulations for the probability of life on a planet?
I'm trying to think of any. The simulators that are easiest to build are the astronomy simulators because they're powered by simple physics and equations. So you'll find a lot of astronomy simulators out there. There aren't a whole lot of geology simulators, and biology simulators are even messier. So if you're looking for the probability of life on a planet, you'll probably want to use something like the Drake equation. So I did build a Drake equation simulator. Unfortunately, I don't think it's open access outside of this course through Arizona State University. But building a simulator that calculates the Drake equation is fairly easy because you could just multiply all those probabilities together. What I did for that particular simulator is I had a field of 100 stars, and depending on what you set the Drake terms to, some of these stars would light up with radio signals. But what I really wanted to emphasize to students there was your numbers have to be really high because you're looking at a very tiny patch of the sky. If you want to have a 1 in 100 chance of spotting a communicating civilization, your Drake equation terms need to be really high. And I designed that simulator as a way of showing students that it's not unreasonable that there are communicating civilizations out there, but we can't see them because we can't see everything.
Oh, okay, thank you. And now another question from Janan Oğuz. How can we adjust gamification and role-playing practices for younger students like primary school students? And do you think it can positively affect children to understand science more effectively?
I think so. The key to creating a useful simulation is to simplify. As scientists, we have a desire to explain everything. And that's where I'll bring in the storytelling elements. Figure out what story you want to tell and only include the plot elements and the characters that you need for that story. So there was a lot of stuff we could have done with plate tectonics and the inorganic carbon cycle simulator, but I focused only on the inorganic carbon cycle because that's what we were teaching. So for younger students, you'll want to streamline that down even more. The fewer things there are there for them to press, do one thing and do it very well. You don't need to do everything. That's the mistake a lot of people make. They try to do everything all at once. Sometimes it's better to do one thing very well than to do a lot of things poorly. And I do think it would positively affect kids because kids are natural explorers. So if you give them a digital toy to play with rather than a video game with a storyline, but you give them a toy to play around with, they'll poke and prod it in ways that can help them discover how these processes work.
Okay, thank you. And now our last question from Melike Naz Shayan. I wonder, we always look for life form that we know in the universe. If there is a life form that we have not known in the rest of the universe, how can we understand it? What are the research about it?
That's a good question, and that's something I actually brought up with some of my students this year in one of my classes where we're doing a CubeSat course. There are known knowns, known unknowns, unknown knowns, and unknown unknowns, which sounds confusing, but there are things that we know, and we know them. There are things we don't know, and we know that we don't know those things. So those are things that are fairly easy to deal with. Then there are your unknown knowns. So there's something that you should have known, but you didn't know that you should have known that, which can be revealed through research. And then there are your unknown unknowns. There are things we don't know that we don't know. So when you're talking about looking for life elsewhere in the universe, we kind of stick to the, well, if you're talking about looking for things that are like silicon-based life or methane-based life or things like that, you're really talking about unknown unknowns. Those are very difficult to explore because we don't know what to look for. So that's where I go back to my conceptualization of science with observations and assumptions. You need to work with your observations and make reasonable assumptions from those observations. When you start talking about life that is that exotic and that out there, you start building assumptions on top of assumptions on top of assumptions, and that's very unhealthy for a scientific model. Because if one of those basal assumptions is incorrect, then all your other assumptions collapse. So when you're searching for this really, really exotic life out there, I know it's exciting and we want to look for things like that. And it's fine to think about it and dream about it. But the reason is that it's not seriously funded is because, assuming we have methane-based civilizations requires a lot of underlying assumptions. And if even one of those is incorrect, then everything else falls apart. So you've wasted time looking for methane-based civilization if the underlying assumption that you can have a methane-based biochemistry is incorrect. The way we understand it is we observe. If we do find something else out there, we need to observe it. We need to make assumptions, and we need to test those assumptions. And anyone can do that. You don't need to be a scientist to do that. Scientists have just professionalized that process that we do every day in making decisions and reacting to our environment.
Okay, thank you for being a part of our conference. It was an amazing presentation, and those were precious information for us. I hope to see you later. Dr. Leo, have a nice day. Bye-bye.
Thank you very much. Bye, everyone.
I'm so sorry, but unfortunately, we have run out the time of our conference for today. Thanks to all audience who watched us today, and thanks to my whole team for preparing such an amazing conference. I know you all are tired, but I hope you are enjoyed also.
Yes, we believe we put out a learning experience that is so unique and effective that is not something that cannot be easily found in Turkey, but also among a lot of student organizations in the world. We try to approach this wonderful field from every different aspect in the most quality way.
Thank you guys for listening and commenting. And also, we all thank you as a team for your kind comments and words.
Thanks to all our audience and Agenastro team. See you tomorrow for second part of the conference.
Okay, see you tomorrow in the second day of our conference, which will be held most in Turkish. Goodbye.